Okay, good morning, good afternoon, good evening, good night, however you're watching this. This is our third lecture, and the title is cell membrane diffusion, osmosis, and it'll end up with mitosis, okay? Now, as you know before, my picture is going to be here, and I'm going to narrate these slides as they come up. This is on your syllabus. It's next to the topic. It says P is in Paul. That stands for PowerPoint, which means you can see this on Canvas, and you'll see it two ways in Canvas. One, this is the lecture. I think it's week one, either the second day or third day. And you can also see it on DocuShare. Now, for those of you who've been around Delta for a while, you know that we used to use, for PowerPoint, we used DocuShare. Just prior to that, they were shifting to another program called Canvas. And when we had the COVID-19 came about, and we shift to an online format, everybody had to go to Canvas. So what my team decided to do is put absolutely everything on Canvas so you can do one-stop shopping. This, therefore, is on Canvas. Look for the title. It is also DocuShare portion of it is on there. In other words, they're both the same, but you don't have to go to DocuShare to get it. And if you want to go to DocuShare, it's still going to be the same one. What I would suggest is you download this, either print it off or look at it, go through it, compare it with your book. So in other words, every slide that you see and I'm going to talk about, read about it first. So when you hear it from me, it's not for the first time. You don't have to do it that way, but I'm trying to give you ideas that will make you successful. Okay? Ain't my first rodeo. Okay. Now, let me get to the start of this thing. Might have to move some stuff around here. Hold on. Oh, I think maybe I just go like this. There we go. Okay, this is the title in the textbook: Cell Structure, Membrane, Transport, DNA, Cell Cycle, and Mitosis. So you see we're going to cover a lot of this stuff. And if you looked at the caption in DocuShare, and I don't know if it came over with it into Canvas, but it said this lecture is going to take 2 to 3 lecture segments. Now that's when we're on the regular in contact course, when it's an hour and a half. So this takes me about 4 hours to go through. I'm going to do it all at once, and you can take breaks. But remember, follow your syllabus, follow what we're doing in Canvas, week 1, day 1, day 2, day 3. If you get behind, you're 1 week behind. So you don't want to do that. If anything, you want to be ahead. Okay? And as I told you before when we did the syllabus and the course information sheet, that if you're trying to work or you're a single parent and you've got kidlings around and you're trying to squeeze this course in, you thought being online it would be convenient because you could do it at any time. No, it's not going to work. Okay? We've squeezed this into 4 weeks. That just doesn't happen. About 3 years ago, 3.5 years ago, no, it'd be either 3 or 4 years ago, I was going to go out of the country, my honey and I, and we were going to go to China, which is right behind me is the Great Wall of China. We took that picture. In fact, standing on the steps is my honey right there. Anyway, that being said, the division dean asked me, "Were you going to do summer?" Because I always do summer. And I said, "No, I'm going to take it off and we're going to go to China." She said, "Well, if I made it 4 weeks, would you consider it?" And I thought, 4 weeks, they're all going to fail. I said, "Yeah, I'd consider it." I'd be a little selfish. I wanted to teach the class, but I also wanted to go to China. Anyway, I did it. To my surprise, the success rate was 80%. Normally, it's 67%, right around there. 80%. In fact, if I remember right, it was like 83 or 86% of the people got A's and B's. That just doesn't happen in my class. And so I tried it again the next year. I think it's been 4 years now. I tried it the next year and I thought, let's see what happens if that happens again, or it was just a fluke. This time I was in town and the success rate was almost identical. So a lot of the instructors and I got together and I said, help me out here. I don't know why. So we did it a third time and the success rate was very close, 78%. And I went, okay, put our heads together, talk with other people in other places, online, other colleges. What we found was, because it's 4 weeks, that the people, that's you, the students that take it completely clean their plate of everything else. No social life, no working, no family, barely eating, and they immerse themselves in the course. And when they do that, it's just 19 days of nothing but biology. Well, that's not true, because that's not counting weekends. 19 days is counting weekdays, but you throw 6 more days in there. So you're at 25 days, 24/7 anatomy. So the students that undertook the class, in other words, took the class, knew what they were getting into. A couple of them in the first 2 days left, but the ones who were there bought in. And they come to campus with me. I started at 5:45 in the morning and they would leave at 7:45 in the evening. They do the SI, they would do tutoring, they'd try and squeeze in for other labs. And what it was is they just gave 100%, which you just don't see that in a regular semester class, because you're taking 3 or 4 other classes. Okay, to you. You're going to take this course. You're going to see that we're going to do 1 or 2 chapters a day. We're going to do 1 or 2 labs a day. You're going to have to take the exam on a certain day, on a certain time, because that's the only time it's going to be available for you. The relaxed part of it is you can stop the video clip that you're seeing right now, go to the bathroom, get something to eat, back up, write your notes. So if you're in class, now this is slated for lecture is 2 hours and 45 minutes. That should take you 5 hours to do. Here's why. And I've already told you this on the first lecture. You're taking notes, you're listening. On one side of your computer, you have your notepad. On the other side of your computer, you have your lecture book. And you're going through it page by page. And you're stopping the video and you're reading it. You're starting the video, you're making notes. That's going to double the time. If you were going to sit down and listen to me and see what I have to present to you, and you're going to go all the way through it, you're going to fail. Okay, you're not going to be successful. There's just too much information to absorb and hold on to. Okay, you got to write it. You got to take notes. You got to take and redo your notes, so you can see that I'm lecturing for 2 hours and 45 minutes. Should take 5 hours for you to get that through. You still got a lab. Labs are not going to be that intensive, but it's still going to take you 3 hours, 4 hours. So you add 5 and 4, that's 9 hours. You got to eat, go to the bathroom, and go to sleep. You're out of time, Jacko, and that's no time for studying. That's just getting the material down. Okay, that being said, all I want to do is caution you, because I want you to be successful. Honestly, I do. Okay? If you can't do that, come back and in the fall, as you probably heard, we're also going to be online, but it's stretched out over 16 weeks. So there you can get by with taking more time. Otherwise, spring, we're going to be back in the classroom, old self. You get to ask questions, you see me every day, that kind of stuff. So keep those in mind. If you're not too sure, email me or email Brooke or email Sarah, and we will tell you firsthand. You tell us what your situation is, and we'll try and guide you. But ultimately, it's up to you. Okay? That being said, let's get on to the lecture. Okay, it says cell structure, membrane, transport, DNA, cell cycle, and mitosis. Those are the topics contained in that chapter. Cell structure, we're going to break that down into three things, which really there's only two, okay? Really there's only two. It keeps going out. When I move it too fast, it disappears. Huh? Look, my arm's gone. Pretty cool, huh? Okay. You think of there's really only two. There's the membrane that goes around the outside, and there's the stuff on the inside, okay? We take one of those, the stuff on the inside is called the organelles, which is called the cytoplasm. It's called the organelles. And you've heard of some of those, mitochondria and the nucleus and ribosome and those kind of things. So when we discuss a cell, we talk about the outside of it because it's very, very unique. And the way the cell is set up on the outside makes that cell be able to do what it can do and what it cannot do. That's why skin cells can't do liver stuff, and liver cells can't do skin stuff. You get it? Okay, that's because of the membrane around the outside. We're going to talk a lot about that. Okay, and then the inside of it is cytoplasm. Now, you may have been taught that incorrectly. Cytoplasm is not that jelly-like fluid inside. It's the jelly-like fluid and all of the organelles. That makes cytoplasm. The jelly-like fluid is called cytosol. Anyway, those organelles inside, one of which is the nucleus, where you know where the DNA is at and that kind of stuff. But we're going to take and separate that one out a little bit. We're going to take and do that one at the end because it's so important. Has your DNA, tells the rest of the organelles what they're going to do, yada, 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 yada. Okay? So three things we'll talk about. The cell membrane, which was a phospholipid bilayer, for those of you who already know it. The organelles and what their general function is, what they kind of look like. And the last one, we're going to take one of those organelles to nucleus and talk about it specifically. Okay? Then once we paint that picture, now in lab, I have an actual prototypical cell, and we look at them under the microscope and all kinds of stuff. You don't have that opportunity. Okay? Anyway, then we do transport. What that means is, as you know, a cell is living. Okay? And that means in order to be living, you have to undergo metabolism. You have to bring in nutrients, use them, make waste, and get rid of it. So it's kind of like your own body. You put food in your mouth, you get rid of it on the other end, okay? And in between there, you used it. Well, the same thing goes on in a cell. One thing is they don't have a mouth, they have that cell membrane. And that membrane has little doors in it. Okay, sometimes they're called channels, but they're little doors that allow certain things in and certain things out. Good? So we call that whole process transport. And for those of you that have had a science class before, we know that transport is diffusion, osmosis, dialysis, yeah? Active transport, passive transport, all those terms we'll go through when we're dealing with that topic. And then we do the nucleus, and when we do the nucleus, we talk about DNA. DNA is only found in the nucleus, and it controls everything a cell does. And when a cell does something, you put a bunch of cells like it together, you got an organ, you got a body, you got a person, you've got an organism, and it's all DNA. Got it? And the way DNA holds that, as you know, a computer has all that information stored in ones and O's, right? Ones and zeros. And depending how many ones and how many zeros, you get a word, or you get storage capacity, you get whatever it is. That's the way a computer works. Well, the way your cell works is you have your DNA. Essentially, you have chromosomes or genes that have a certain amount of genetic information that code for a trait. The trait can be blue eyes. The trait can be a nice hairless head. The trait can be making certain antibodies. Got it? That's all tied up in your DNA. So we'll talk about that. We won't go into long depth on that, very deep into that, because if you take biology 1, that's cell and molecular biology, you'll go way into that. And you know, with DNA separation, nucleotide separation, you may have heard it, PCR, That's all dealing with that nucleus and what's inside of it. Okay? Once we hit cross that one, we're going to go to, here's where I'm at. We're going to go to cell cycle. I think probably everybody listening to me right now has heard of the cell cycle. Mitosis, cell replication. You have one cell, now you have two. You have two, you have 4, and 8, 16, 32, 64, 128. That's how your body does it. And that process of making one cell and making it two, duplicating it, cloning it, is called the cell cycle. And every cell in your body is in a cycle. The cycle is either interphase or mitosis. Interphase, we're going to see, is divided into 3 parts, and mitosis is divided into 4 parts. Got it? So the cell cycle and mitosis. Once we finish that up, then we'll go to chapter 3, which is titled embryology. And we don't do the whole thing. The only thing we do out of embryology, that chapter 3, is we do meiosis, making of the sex cell. And I'll do that one under a D, directed teaching. So you won't have this background of the Great Wall of China. You'll have well, I'll be in the lab doing it for you, and I'll be writing on the board, and I'll make sure you understand exactly how it goes. Good? So that's the next chapter after this one. Okay, here we go. Now, this is not your textbook. This is edition 4, and you're in edition 6. I just didn't change it, okay? They update all the material in it. Anyway, here we go. By me. Okay. Topics of discussion. Now, I'm going to make this a little smaller because I don't know if you can read all this. I don't know if you're seeing the same thing I am. But what I'm going to do is I'm going to take and make this me a little bit smaller. Okay, like that. That's good because you're only listening to my voice. I could do this with my voice, but I thought that if you saw me, how good looking I am, you might hold your concentration. Okay, topics of discussion. Now remember, with introduction, I always do topics of discussion with bullets. And I want you to engage yourself. Here's what I'm doing. I'll show you a bullet like this. Study of cells, okay? Now, the study of cells is, there's a topic. There's a Latin word for the study of cells. What is it? Now, remember what we did before. I said, pause it, pause your computer, think. Study of cells. What is study? Ologies. Ology. What is cells? Sight. Cytology, study of cells. We're going to talk about that. A prototypical cell. In lab, I have one. A prototypical cell does not exist in your body. It means a cell that we as scientists or instructors, teachers, have put out there that has everything in it. None of your cells have everything in it. Some of them have a little bit of most of the stuff. Some of them have a lot of most of the stuff, but none of them have all of the stuff, okay? Otherwise, you'd only have one type of cell. So when we have a make-believe cell, we call it a prototypical cell. Got it? And I'll show you what that is. Plasma membrane. Another name for plasma membrane is cell membrane, okay? Plasma membrane, cell membrane. Cell wall, not the same thing, ever, 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 okay? Every cell, no matter where it's at on this earth, has a plasma membrane. But if you're a plant cell, or you're a fungi cell, or you're some bacteria cells, you have an extra coating on the outside called a cell wall. Okay? We won't be dealing with that. But we're going to spend a lot of time talking about the plasma membrane. And I'll tell you why. It's because every group of cells in your body that are the same or very, very, very similar are set up the same way. So they can do the same function, which means they have to have the same nutrition, they have to have the same ability, and they create waste and you have to get it out. That's all done through the plasma membrane. Plasma membrane, when I teach anatomy and physiology, when I do the physiology part, plasma membrane is huge, huge to understand that, okay? Cytoplasm. Now remember I said that there's 2 things in a cell. The outside, which is called a plasma membrane, and the inside, which is called the cytoplasm. And remember I said we were going to do a third one? Okay, I said we were going to do a third one, and that third one is the nucleus, which is still part of the cytoplasm, but we separate it out. Okay, so the cytoplasm is the jelly-like fluid with all the organelles. and then the nucleus. Remember where the DNA is at? Okay, so we'll separate that one out, still part of the cytoplasm. Life cycle of the cell. Remember I told you what that was? Essentially, mitosis. Life cycle is broken down into two parts, okay? I move too fast, it goes away. Okay, it's two parts, interphase and mitosis. Okay, and I'll do that. And that takes us right to the end because in the life cycle of the cell, we'll talk about mitosis. Mitosis is in that little portion. And aging the cell. Most of the topics we will discuss here, I will go into one that talks about aging. Because as you know, and don't look at me, do not look at me that way, that the population in the world is getting older because of a variety of reasons, nutrition and antibiotics and all the information and stuff like that. Life expectancy was 40s, 50s, 60s. Then it was 50s, 60s, 70s. Then it was 70s, 80s, 90s. Now it's nothing for somebody to be 100 years old and still driving a car. My mom's 90 going on 91, and she still goes all around, volunteers, the police department, does all kinds of things, okay? So I'll talk about aging because it's something you can't stop, okay? Plastic surgeons are making a lot of money making you think you can stop aging, but you still start sagging and everything else, okay? So I will talk about aging. I will not talk about embryology, okay? I won't talk about that stuff because that's a separate course called embryology, and you'll take that when you do probably upper division if you go on to get your bachelor's. Okay, here we go. Study of cells. What did I say it was? Pause. Pause your computer. What did I say the study of cells was called? Okay, got it back on again. Here we go. Cytology, study of cells. Will I ask a test question on that? Probably. It's going to be easy. Either you know it or you don't. If you know it, big deal. You get one right. Missed the other 99, but you got one right. Okay, cytology, only visible by microscopy. That means in order to see a cell, you've got to have a microscope. If you look at your finger, okay, your finger is made up of hundreds and thousands of cells. Can you see any of those cells? Can't, can you? Which means you need the help of a microscope, okay? Cells are major, now we did the metric system, didn't we? So you know what micrometers are, and that's how we abbreviate it. Micrometers, remember meters, lowercase, okay? This one should be, remember, no, it's right. Okay, so it's measured in micrometers or micrometers, same thing, just you accent it differently, okay? Now I put this one in here, kind of like a little practice. Remember what went above the micro sign? You should be right now, pause, think. Okay, now we're back with me. You should have put a 6 up there, didn't you? Didn't you put a 6? And what goes above centi? 2, right? 2, right? Yes. What's the difference between 6 and 2? 4. How many zeros do you see? 1, 2, 3, 4. And there's a 1. You see? So 1 centimeter equals 10,000 micrometers. You did that conversion. There was 15 of them on your to practice with, huh? Okay, that's why I had you do them. Okay, that's why I left that one in there. Sizes vary. Okay, does every cell in your body, is it the same size? The answer is yes, no. Most cells are the same size. Now they have different shapes. A nerve cell is like a piece of thread. A blood cell is a circle, a cylinder, a sphere. Your skin cells are kind of like squamous. They're kind of flattened like a fried egg. Kidney cells are cuboidal. They're like a cube. So we have different different shapes of cells. Pretty much the cells are all the same size because we have an exact surface area to volume ratio. Remember we did that in the metric system. Remember, the surface area is 2 things multiplied together. That means your answer is squared. the volume is three things multiplied together. So volume is cubed. So when something is squared and the volume, so the outside is squared, the inside volume is cubed. It's getting bigger faster than the outside, and it becomes inefficient. You can't feed the inside with the outside because there's not enough outside to go there. So you get to a certain size. And what I do, and when we're in contact, I said, picture a flea. Okay, if you don't know what a flea looks like, it's something really small. And an elephant. If you don't know what an elephant is, I'm sorry. Okay, but an elephant's very big. Are their cells exactly the same size? And you think, well, hell no. Elephant's got to have bigger cells. Wrong. It just has more of them. The cells are exactly the same size because of surface area to volume ratio. But now getting back to the thing, it says sizes do vary. A red blood cell is the smallest cell in your body. It's also the most common. And a female egg, which we call an ova or ovum, is the largest. Okay, so there is a variancy in size of cells, but pretty much they're all the same size. Okay, so I put 7 microns or 7 micrometers, which is a red blood cell, to 120 micrometers, an oocyte, which is the egg. Okay, I'm going to ask you that on an exam. Okay, shapes vary. Now we just talked about this, depending on the location and functions. Shapes of cells are different. If all the cells were the same shape, you'd only have one cell. Figure that out. Okay. All right. Flat, cylindrical, oval, irregular shape. Those are all cells in your body. And you'll know that because we're going to do tissues and you're going to actually look at it under a microscope. You're going to see a picture of it. Okay? And this little one right here, I said, you know, we remember when we were doing the oh my gosh, what did they do here? Okay. We said the atom is the smallest thing that's not divisible by a force. Okay, and that's the periodic chart, you know. And then we put small molecules, okay, like oxygen and carbon dioxide, and macromolecules. Remember, there was 4 of them: carbohydrates, lipids, proteins, and nucleic acids. And then we talk about the size of viruses and ribosomes. And boy, we've seen that virus all over the place, huh? Like the COVID-19. And then we talk about the organelles, mitochondrion, then a red blood cell. See what we're doing, getting all the way up big, you know, 1 meter. 1 meter is 3 feet. This pretty bigger, 3 feet. We good? Good. So it just says size. Now here's an oocyte, and there's a red blood cell. Okay. All right. Types of microscopy. I remember when I sat in your seat, science was, because we were starting to look at things very, very, very small, we had microscopes and people were trying to find the best microscope to use to tell certain things. So there was all kinds of microscopes. There was phase contrast. There was a compound light microscope. And what was coming along really quick was electron microscope. And here's the reason why. We use a compound light microscope. You've seen microscopes. You look through a microscope. That's called a compound light microscope. Light is the source to illuminate. And compound means you have 2 or more lenses. And there's 2 of them. One in your eye and one down by the specimen. Got it? So what has happened in science now, because we've gone, you know that we have seen proteins. We can look under a microscope and see a protein. We can see a mitochondria easy. Okay? But I take you back 100 years ago, they couldn't do that. They didn't have the electron microscope. And the reason the electron microscope can see things so small is, picture this. Light comes from the sun in little packets, like little pillows of energy. And those pillows bounce into each other, and there's a space between one pillow and another. And that space, depending on where you're at, if you're standing on the surface of the earth, you're up in the atmosphere, those spaces get larger and smaller. So when the light travels to your eye, our eye cannot see small enough, so we see a complete room of illumination. But when we start looking at the microscope, when we magnify, in other words, using the lenses, we magnify it. We also magnify the spaces in between that are dark. So we could actually put lenses on a compound light microscope that would magnify something 1,000,000 times. but it will also magnify the darkness a million times, and you wouldn't be able to see anything. So we can max out compound light microscope. We use electrons. Electrons can be put, almost smash them together. So it's almost endless. Got it? Okay. We have found that there are two types of electron microscopes we use. We have them here on campus. But the first one we're talking about is electron microscope, abbreviated LM. I'm going to have a test question on this. Okay, the reason we use light microscopes is because they're cheap. Yes, even though they cost $2,500 for a good one, which you'll see it you will see this if we had face-to-face, but in micro you will. They're relatively inexpensive. Electron microscope, couple million. Okay, we can't give you guys each one of those. Okay, maybe in another world we could. So light microscope's cheap. We get to see things, but we can only magnify it about 1,000 times. And then it starts, remember, magnifying the darkness. Visible light passes through the cell. Okay, we don't get to see the outside. We can only see the inside because we have to slice it like a loaf of bread. We'll use one in lab, sorta. Transmission, or transmitting electron microscope, abbreviated TEM, transmitting electron microscope. You're always going to see electron. Transmitting electron lets us see the inside. Okay, so when we want to see the inside of something, and we want to really magnify it, we use a transmitting electron microscope. Have my secretary get that. Okay, she can get off the couch. A beam of electrons passes through a thin slice of a specimen. So you're thinking, wow, that's pretty cool. If that's what I want to use, I would too. But the only thing is you got to slice it. Okay, try slicing a piece of bread a micron thick. Okay, obviously it's going to be very tricky. Okay, so that is a disadvantage. You have to slice it. Another disadvantage is you only get to see the inside. You can't see the outside, which brought us to the next type. Scanning electron microscope. By the way, transmitting is two-dimensional. You only get to see the inside. Like when you slice a piece of bread, you don't see three-dimensional, you see two-dimensional. Scanning electron microscope, you don't have to slice it and you see the outside, which is pretty cool. So when you see pictures on TV of the COVID-19 virus, okay, what you're actually looking at is through a scanning electron microscope. You see those whole things sticking out there? Those are proteins. Okay, we're going to talk about that. It says beam of electron bounces off the surface of the cell to provide a 3D image of the cell surface. Pretty good, huh? Pretty good. All right, now the next one I'm going to show you is a picture. Now remember how we did it before? We said, first thing I do is give you an outline, tell you what's coming. Then I take each one of those bullets and we talk about it. Talk, 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 and then I show you a picture. And if I have a table, I show you words, picture, table. Got it? Okay, in this case here, the three types of, I'm going to show you a picture of a cell or a portion of the cell using all three different types. Okay, ready? Here we go. This is a compound light microscope. This is ciliated pseudostratified columnar epithelium. I know that's a mouthful, but you'll learn it here in about another 3 days. It's got cilia on it. The shape of the cell is columnar, and the layers is pseudostratified. Pseudo means false. So it kind of looks like it's layered, but it's not. If it's not layered, it's simple. If it's layered, it's stratified. But one that tricks you is called pseudostratified. And that's, we find this picture right here in the respiratory tract. That's your nose, lungs, throat, that kind of stuff. And you can see that it's a light microscope magnified 720 times. That's pretty darn good. Now, that means with our microscopes, we can see something not quite this magnified. We see it about 400, 450 times magnified. Well, that's pretty good, huh? And then we all have our own microscopes. That's the advantage. Now here's a transmitting electron microscope. You see this, and that are the same thing. They're just really big. Look at magnified 50,000 times. And we can actually look inside and see the proteins. So we know that cilia, there, and there, is made out of proteins because we can see it. Well, I don't know what the heck happened, but that didn't look good. Okay, there we go. And you can see how these pieces broke off. That's because when they made the slide, they had to cut it. Remember that? And it broke off. So these things are all the same thing. And this is a scanning electron microscope. Now the cilia that you see here and here is used to filter air, to get all the impurities out, the dust and that kind of stuff, and warm it. Does that look like it can filter anything? I hope you would say no. Does this look like it could filter anything? I hope you would say no. Does this look like it can? Air is traveling through here, in and out and all around. You're thinking, well, heck yeah, I can see that. Looks like a sea of grass, huh? Okay, do you see the advantage? This one, everybody gets to see it. This one, we can see what a protein looks like on the inside. This one, we actually see it on the outside. Got it? Pretty cool, huh? Pretty cool. Cellular and tissue functions. Okay. You know we have a cell, and like cells, L-I-K-E cells, form tissue. And like tissue, L-I-K-E tissue, forms organs. Organs working together is a system. Remember that one I showed you? The dude standing with a cup of coffee? And then systems working together makes an individual, an organism. Okay, got that? Okay, now, what is tissue and cells used for? Coverings, to cover our body, to cover the inside of organs, and we use it on covering on the outside and on the inside. That's lining. When it's on the inside, we call it lining, okay? Inside's lining, outside's covering. Storage, oh my gosh. We know we have cells for storage and tissue for storage, don't we? Don't we? We're always trying to shed that storage, aren't we? Huh? I finally got myself down to, I'm not going to tell you, but I was working at it and working at it and working at it. Finally, I got it. And man, it's so hard to pass up the pastas and stuff. But if I don't pass them up, I know my body's going to reach out and use them as storage. And hence we have lipids, don't we? Some people call it fat. Okay, so we use for storage. Okay, movement. We use tissue for movement. What kind? Pause. Okay, now you're back with me again. Movement, muscles, huh? Muscles cause the skeleton to move, which allows us to move. Okay. Connection. Now, you know that our whole body is connected with like a glue. If not, you could take your skin and roll it all the way around. Okay. When you made a hard right turn, your intestines and everything would go to the left if they weren't glued in there. So we have tissue that does connection. In fact, that's what it's called, connective tissue. And then defense. We know about our white blood cells. Huh? Something comes into your body that's not supposed to be there, your white blood cells go to work. And that's your defense. We do it a couple ways. We can do it by cell eating, phagocytosis, or we can do it by creating antibodies. We'll talk a little bit about that. Communication, the nervous system. Boy, are we going to spend some time talking about the nervous system. That sucker's complex. Okay, it's just not the brain and your spinal cord. There's nerves and there's nerve cells and there's helper cells and there's all kinds of stuff. So we get to the nervous system. Hang on. And then, oh boy, do we know about this guy? Reproduction. Yeah, we have to have cells that their job is just to reproduce. Now, I don't mean making babies. I'm making half a baby, a sperm and an egg, and that's meiosis. And at the end of this, the next lecture is on meiosis, and I'll tell you how that's done. Are we good? All right, here we go. I don't know what that says. Oh, here's pictures of it. Remember? Words, pictures, tables. So here's the cell that you're going to be able to identify for the lab exam. Here's skin, stratified squamous. Here's intestine, simple columnar. And here's muscle, and here's sperm, and here's nerve cells and blood cells, and yada, yada, yada. Got it? You'll be able to do this, or you're not going to do very well on the exam. Okay? Anyway, that's just a picture to show you what we're doing. Prototypical cell. Remember that one? Prototypical cell was a cell that doesn't exist, but we, as scientists, put it all together to give you an example, huh? I'm looking, you're right. I am looking at my cell phone because somebody called and I said, oh, is it important? I have to address it, right? It's not. It's one of my cousins. She sends about 40 of them. She's very bored in her life. So she sends pictures of stuff. She sent me a picture of a friggin' tomato, like I've never seen a tomato. And then sends me a picture of her flower, her favorite flower. Now, she's a nice person. Don't get me wrong. But I got other things to do in my life than look at a friggin' flower on my cell phone. Anyway, prototypical cell. Now we said nearly all human cells have three basic parts, really two: the outside cell membrane, inside cytoplasm. And then we separate the last one out with a nucleus. Remember that? Okay. Plasma membrane, that's the outer barrier, and it's selective. That means it allows certain things in and certain things out. That's what the word selective means. Are we good? Cytoplasm. Cytoplasm is that cytosol, the jelly-like fluid, and all of the organelles, including the one that's going to pop up the third one, which is the nucleus. Are we good? Okay. Okay, cellular contents, cytosol, organelles, and inclusions. Scientists always do that, when they miss, inclusion. So we have, we have a ribosome, and we have a Golgi body or Golgi apparatus, and we have endoplasmic reticulum, and we have some other stuff, some proteins and some other stuff like that, ions and amino acids stuff. Those all fall into inclusions. Got it? Okay. And then the nucleus. Do you notice how in blue I put part of the cytoplasm? I want you to realize it's part of the cytoplasm, but realize we're going to separate out because of that old DNA thing. Okay? It's the control center. Okay? Whenever the cell needs to make another cell, or it needs to know what to do, or you have an infection, it goes to the nucleus, says, hey man, what do I do? Like the hard drive in your computer. Okay. Structure of cell. Here's a prototypical cell. I'm going to go over some of these for you because I'll refer back to it. I'll say, hey, do you remember that blue thing? Remember this thing inside the nucleus? Remember the thing that looked like a hot tamale? Wait a minute. I got a hot tamale. Just a minute. Still working on it. I want to get one at a time. After I show it to you, I want to eat it. Okay. Remember the old hot tamales? Candies? How about cinnamon candies? Okay. You got a hot tamale? You don't see it. I'm going to go like this. Go like this. You can't see it. It's just still moving too slow. Look back. There it is right there. See it? Doesn't that look like it too right there in that cell? Look at the one down inside of here. Look at a hot tamale. That works for you, great. Okay. Anyway, the little hot tamale thing's mitochondria. These are centrosomes, centrioles. They help in division, cell division. This is a Golgi body, Golgi apparatus, Golgi something. Okay. Okay. This is endoplasmic reticulum. Find out what endoplasmic reticulum. There's two types, smooth and rough. We'll talk about that. This is the nucleus. The nucleus, and those holes are not called nuclear holes. They're called nuclear pores. P-O-R-E-S. And this is supposed to represent DNA or chromosomes or chromatin or whatever you want to call it. We'll talk about that later. These things here are called transport vesicles. So what essentially happens is proteins are made in this gold thing according to what this nucleus tells it to do. Got it? And then it travels in these. These are a series of tubes and channels that lead to the Golgi. In the Golgi, things are separated, packaged up so they're all the same. So you don't have 20 different proteins in one little basket. You have 20 baskets with each having their own proteins in it. And then it bags it up in a vesicle. That's what it is. A vesicle is like a bag. Okay? And all the proteins are the same. And then it goes over here and it dumps it outside. See that? Because of the plasma membrane. So we call this type of vesicle that's dumping something outside a secretory vesicle. S-E-C, secretory, R-A-T-O-R-Y, secretory vesicle. Okay, because it's secreting. Some of them stay inside. We call them a lysosome. Good. Okay, now you see some things hanging out. I just ate a mitochondria. These are called microvilli. We only find those in the intestine. And that's the slow stuff going alongside the cell, slow enough to where it can be absorbed back into the cell. Otherwise, it just slip right out there and out your backside. Taller ones are called cilia. We saw that. Remember the ciliated pseudostratified columnar epithelium, Remember the ones I said in your respiratory tract? Well, this is what they look like. And you know what they're made out of, huh? What's that little green thing in there for? That's to represent protein, huh? And then we have in the male sex cell, which is sperm, we have this long guy here called a flagellum. And that's for movement. Flagellum is for movement and cilia is for movement. But if the cell is anchored, that can't move the cell because it's anchored. It's used to move things by the cell, past the cell. Got it? Got it? Here's smooth ER, here's rough ER, mitochondria, lysosome, secretory vesicle, centrosome, et cetera, et cetera. We're going to go over each one of those. Ready? Here we go. I have another mitochondria. Plasma membrane. Now we're going to talk about uniqueness of the plasma membrane. Remember the plasma membrane, cell membrane, same thing. Also called the cell membrane. It's a phospholipid, and there's two layers called bilayer. Sometimes they put those words together, a phospholipid bilayer. Okay? Now, you know what a lipid is, fat. Okay? You see that a lot in Costco. Okay? You see fat. Whoops, lost it here. A lipid. And it has a phosphate. That's on a periodic chart, a phosphorus. We call it a phosphate. Phosphorus molecule attached to a lipid makes it a phospholipid. And that's what all cell membranes are made out of. And there's two layers of it. That's why I get the word bilayer. But it isn't a solid band of phospholipids, not a solid band. It has things smashed in between it, okay? And the things that are smashed in between it are called, another mitochondria, are called proteins. And we call them protein gates, proteins channels, those kind of things. We'll talk about that when we do diffusion. So it's selectively permeable. Read all this, it tells you. Selective permeable means it's going to let certain things in and certain things go out. Got it? All right. It says, what is a plasma membrane, cell membrane made out of? The main thing, phospholipid, huh? And two layers of it. Two layers of it, phospholipid bilayer. And I said that there's also channels and gates in there made out of proteins. So it says the main components are that. Good. Lipids. Okay. What do I have after that? And the major lipid is a phospholipid, and it's a bilayer. Good, good. Got it. Cholesterol. We've heard good things and bad things about cholesterol. So, oh, you can't eat too many chicken eggs because it's too high in cholesterol. And cut your cholesterol down, and HDL and LDL, right? High density lipids, lipoproteins, cholesterol, all those things. You can't have those things because it'll clog your arteries and stuff. Well, what the heck is, you got to have cholesterol, Jacko. You just can't flood your body with it. And what cholesterol does, it is a major component in hormones. So in other words, you start with cholesterol and you add things to it, it makes different hormones, but you got to have the cholesterol, okay? If it's a lipid-based hormone, otherwise it's a protein-based. And the other thing is it holds, remember that phospholipid bilayer? Remember that phospholipid bilayer? Two layers? Well, what holds that layer together is cholesterol. Wow. So now we know we have to have cholesterol, huh? Okay. Glycolipids. Now, when you see this, don't panic. You see this lipid right here? See a lipid right there? This lipid right here, the phospholipid, had what attached to it? Phosphate. This lipid has a carbohydrate. And so we don't call it carbohydrate lipid. We call it a glycolipid. That's what it is. It has a molecule of glucose or a molecule of a carbohydrate attached to it. Okay, now that is in the phospholipid bilayer. So I'm talking about what is in a plasma membrane. Okay, lipoproteins. Now look at it again. What do you see here? Lipo and protein. So we have this macromolecule, a lipid, and we have a protein attached to it. Here we had a carbohydrate attached to it, huh? Here we had a phosphate attached to it. You getting it? So we're going to have a bunch of protein and lipids embedded in it. We're going to have a bunch of carbohydrates and lipids embedded in it. You see what I mean? And embedded in it being a phospholipid. You understand? So it makes it act differently. And then proteins. When things won't go through the phospholipid bilayer, and I'm going to show you a picture in just a minute. When things won't go through a phospholipid bilayer, we have to put a bigger door in. We have to. Can you get through a wall? Oh yeah, if I run fast enough. Come on, be serious. Can you get through a wall? You can't, can you? But can you get through a door in a wall? That's what it is. The proteins are doors in the wall. Okay? Although we don't call it a wall, we call it a membrane. Good? Now there's two types of proteins. Integral. What does integral mean? It's integrated, inside to outside. Remember we have two layers? Okay? When the outside and the inside are connected by a protein, it's called an integral protein. If the inside is connected to the outside by a protein, it's called an integral protein. If the protein is just like a doorknob out there, just something to grab onto to get inside, it's called a peripheral protein. Okay? It can be attached to the outside and the inside, just like your door. Does your door have a doorknob on both the inside and the outside? Does that doorknob say you can go in? No, but you have to twist the doorknob or hold on to the doorknob to get inside. The door is called the integral protein, peripheral is attached to it. You get it? Okay. Picture. Okay. Now, do you see all the purple balls? Let me see if I can. See all the purple balls and the little yellow legs? That is a phospholipid. The yellow legs are the lipid. The phospho or phosphate is the purple ball. And you can see there's one layer here and one layer here. And guess what this is holding them together? What do you think? Come on. We talked about it. Pause it. Look at your notes. You're taking notes now. Look at your notes. Oh, yeah, cholesterol. There's the word. You happen to read it. Cholesterol, that's what's holding that thing together. So therefore, you need cholesterol in your diet. Hello? If you want to sell, you sure have to. Now, they show these things that go here, this blue and green. Those are your integral proteins, okay? And what they do is they show you, instead of showing you all blue ones or all green ones, they show two different ones to make sure. They could have put purple and green and yellow and orange and black and all those other colors. They just put two to say there are different proteins, okay? So this protein may allow amino acids, and this one allows monosaccharides through, you see? And these things that look like tree limbs, okay, those are glycolipids. Remember that? The carbohydrate and lipid, together we call them a glycocalyx. Here it is, glycolipid, you see it? Because the glyco, the sticky part, the sugar is attached to a lipid, glycolipid. Okay, it's part of a glycocalyx, which means all these little arms, whether they're going to a protein or whether they're going to a lipid or whatever, is a carbohydrate. It's a part of glycocalyx. Got it? And it talks about a glycolipid, and it talks about a peripheral. Do you see this? See how this doesn't go all the way through? There's your doorknob. There's your door. There's your doorknob. There's your door. This door didn't need a doorknob. It's a swing both ways. You just touch it, it goes open. Don't need a doorknob. So what's the use of a doorknob? Well, makes it more specific, doesn't it? You have to have the right hand or right key or whatever it might be. It makes it more specific. So if something's traveling along here, it wants to get inside the cell, and it has to attach to that, and that's not there. It goes right past this onto another cell. Okay? If it's inside the cell and it wants to get out, it has to attach to that. You get it? Selectively permeable. See all these little lines? Okay, these are proteins. They're called a cytoskeleton. You have a skeleton? Come on. You saying, well, yeah. Okay, good. That wasn't a hard question. You have a skeleton. Yes, it's made out of bone. The cells, because they have shape, they have a skeleton. It's not made out of bone, made out of proteins. That's what those are, cytoskeleton. And all this together, all this stuff in here is called the cytoplasm. The jelly-like fluid, cytosol, peripheral protein, integral protein, cholesterol, lipids, phosphate, phospholipids, glycolipid, lipoprotein. You get it? Get it? And you can read this stuff down here. Phospholipids. Okay, you see that? What do you think of? Come on, we just did it. What do you think of when you see phospholipids? You think lipid with a phosphate group attached to it. What color? Remember, purple with the little yellow legs. Yellow legs were the lipids, which we're going to learn is fatty acid, and the phosphate was purple. Yeah, most common structure in the plasma membrane. If I laid out 500 plasma membranes out here and I melted them all down, the most common thing you'd see would be a phospholipid. Kind of like a no-brainer, huh? Go back to that picture. What'd you see the most of? In fact, I'll back you up to that. What do you see the most of? Purple balls with yellow legs, huh? Phospholipid. You also see proteins, yeah, but not that many of them, do you? Okay. So first thing, most common structure in a plasma membrane. All right, now we're going to get into some technical stuff. So far, what I've been teaching you, I can teach to a 5th grader. Now we're starting to bring you up to that level. It says polar and non-polar. Okay, if something is polar, water can touch it. If something is non-polar, water cannot touch it. Do you understand? Your blood is the major component. 55% of your blood is mostly water. Okay, mostly water. That means if a cell has blood going around it, which would make sense because your capillaries are carrying blood and carrying the nutrients there, they got to be able to touch. That means phosphate is polar. It can touch water. The fat, you know, when you put fat in, you got a bowl of water and you put some oil on it, what happens? Stays right on top, doesn't it? It repels it. You've heard that term, hydrophobic. Yeah, I'm scared of water. So that's nonpolar. So the fatty acid, the lipid part, is faced inside. So the inside of the cell contents and the outside of the cell contents can touch water. The lipid in between can't. That's why we have that protein going all the way through, don't we? The integral protein. Forms a phospholipid bilayer. Okay, we just did that. Polar regions, exposed to water. Didn't we just talk about that? And you have polar regions on the outside, you have polar regions on the inside. That's why you have those purple balls on the outside, the inside, with the yellow legs in between. Make sense? Non-polar regions face the inside. Okay, that's the yellow legs. That's the lipids. That's the fatty acid. Other lipids, cholesterol. Remember we talked about that? Cholesterol is lipid-based, and we use cholesterol to hold the phospholipids together. We use them to make some of the hormones we use in our body. Stabilize the two layers together. Don't worry about the second bullet, that's stupid. Strengthens and stabilizes membrane against extreme temperature. I don't want you to worry about that. I want you to know that it holds the two together. And we have components of that. Glycolipids, that's a sugar lipid. Lipoproteins, that's a protein lipid. Glycoprotein, that's a sugar protein. You see it? Remember those 4 macromolecules we talked about? Carbohydrates, proteins, lipids, and nucleic acids. Let's take nucleic acids. We don't worry about that right now. We're going to do that when we do the nucleus. But look at these 3 guys. Okay, so we see they're combinations of those 4 that I just mentioned, huh? Because here's lipid, here's lipids, here's a carbohydrate, here's a carbohydrate, there's a protein, there's a protein. You see it? You see it? You're going to need to know those test questions. They have a carbohydrate sugar molecule. Let's tell you, we're talking about a glyco something. Okay. And the glycocalyx, remember that? Okay, it's a name given to all of those. These things right here are all 3 separate things. but together we lump them into a common title called glycocalyx. Cell-to-cell attachment. Now, what holds cells together? You can't wish a cell together. They got to be attached. Now, you know that the outside of each cell has a phospholipid bilayer, correct? Got it? So that means if it has a bilayer, that bilayer has to have something in it to make it stick. Have you ever gotten sugar on your fingers? Sticky, huh? So glycolipids and glycoproteins, the glyco part is sticky. Now that's a weak cell-to-cell attachment. What really holds it together strong is proteins. And we'll talk about that. Cell-to-cell attachment, strongest thing is protein. And they signal the cell type. Hey, can you take an eye cell out, put it in your finger, and it becomes a finger cell? No. How come? Because it doesn't have that signal type. When the eye cell comes out, it says, hey, I'm an eye cell. When the finger cell comes out, it says, hey, I'm a finger cell. You can't put the two together and says, hey, you work it out. It didn't work that way. Okay? So signal the cell type. That's what that glycocalyx does. Signal what type of cell we are talking about. What's next to it? Okay, remember the membrane proteins? What kind were there? Come on. There was two types, weren't there? Think, pause, think, go back through your notes. Okay, you pause me, now you're back to me and you found the answer was integral and peripheral, huh? Okay. Proteins are made of amino acids. So you take amino acids, you put them together. By the way, we have 20 of them. There's only 20 different amino acids in the whole wide world. Okay? And you take those amino acids and you put them together, depending on how you picture it as a pearl necklace. Okay? All pearls are a little bit different. And depending on how you put the pearls together, you get a different type of necklace, don't you? Okay, so depending on how you put your amino acids together, you get a different type of protein. There you go. Okay, two types of membrane proteins. You already know this, integral and peripheral, right? Integral goes all the way through. Peripheral is the doorknob, either on the inside or the outside. Are we good? Not now, because I'm going to start picking up the speed, it's not because it's any less important. You can read this, pause it, read it, write it down. Good? All right. Integral proteins. What do they do? Pause this. What do they do? Pause it. Okay, you paused it, you came back and you found integral proteins go all the way through the phospholipid bilayers, don't they? They go from outside to inside or inside to outside, correct? Go back to our prototypical plasma membrane. Remember the purple balls and the yellow legs? And then on the other side of purple ball and yellow legs, and then remember the green and the blue? They went all the way through, didn't they? And some of the green and blue things had a yellowish gold thing attached, which was a peripheral protein, the doorknob. Okay. Integral proteins embedded in the phospholipid bilayer span the entire thickness of the membrane. Go back to the picture in your mind. In fact, you know what this would be good is to go to your book, take the picture that you saw that I had, the phospholipid bilayer, make a copy of it, make a color copy. Yes, you can. Yes, you can. Make a color copy, make it a little bit smaller, and put it in your notes on the opposite page. That way you can look at it while I'm talking right now. Pretty cool, huh? It says expose the outside to the inside, inside to outside. Uh-oh, another name for it, okay? So we don't only call them integral protein, we call them transmembrane proteins. This is kind of like when you get into heavier, what am I looking for? When you get into talking about cells more and cytoplasm more and what the function of those cells are and that kind of stuff and DNA, they call it transmembrane, but we know it as an integral protein, okay? Integral protein, transmembrane proteins. Still the same thing, still the same thing. Cell and molecular biology is what I was thinking. Just didn't come up with it. It says you can have carbohydrates attached to the outer surface, making them glycoproteins. We already talked about that, okay? All together, they're called glycocalyx. Remember those three things together are called glycocalyx? There you go. You have that in your notes. Go back one slide, there it is. And it says it has many varied functions. I had to put that bullet in because that bullet is in your textbook. That's pretty stupid, because if they had all the same function, you'd have one type of cell, wouldn't you? Okay, if they all had the same function, you'd have one type of cell. Peripheral proteins. That's the doorknob. Remember the one on the outside, kind of goldy shallow? Not embedded in the phospholipid bilayer. Not embedded in the phospholipid bilayer. Okay, it's attached to the external or internal surface of integral proteins. You already know that. Has many varied functions. Now remember, I had to put this in there because it's in your book. But of course it has varied functions. Okay, good? All right. Example, enzymes, cell-mediated attachment sites. That's how they work. Okay, got it? Proteins similar. Oh, what do we do? What are the proteins used for? Remember the green and the blue? Remember the green and the blue? Integral proteins. What are they used for? Duh, transport. Getting things into and out of the cell. Good? What else? Intercellular connections. Remember? I said weekly we use sugars, glyco. We use those to kind of cell-to-cell recognition. Kind of, okay, that's how I'm going to tell you who I am. And we kind of like hook up. That means not that kind of hookup. Okay? But proteins are what really holds us tight together. Okay? It's like putting two arms in one sleeve. Okay? One arm sleeve of a shirt, you put two arms in there, baby, you ain't going nowhere without that other person. Make sense? Okay. Anchorage for the cytoskeleton. Remember those lines I showed you and I said they're made out of proteins? Remember in the prototypical cell? No, it was in the phospholipid bilayer. And we had those little lines floating through there. Okay, those are proteins, and the phospholipid, the proteins attached to that, give them shape. So anchorage for the cytoskeleton. Hey, what's the difference between a catalyst and an enzyme? Or is there a difference? Pause it. Pause it. Okay, look it up. Look it up. Pause it. Look it up. Okay, you didn't look it up. I know you. I know you're waiting for me to tell you the answer because you knew I would. If a catalyst is something that makes a reaction happen, the way the books say it, it lowers the energy of activation. Okay? In other words, it takes energy to make two things come together. You take A, B, they just don't become C. Okay? A and B take energy to put them together, smash them together. When you use a catalyst, it takes less energy. With the catalyst, kind of gives it the extra energy to do it. Okay? So here's an example. If you have a water and you pour Kool-Aid in it, what will happen to the Kool-Aid? It'll sink to the bottom, sit there. Some of it dissolves, some sits there, right? Okay, that's A and B. But if you start to take a spoon and you start swirling around, pretty soon all goes into solution, doesn't it? Guess what the spoon was? Catalyst. Okay? And now, when I was young, we had what was called ghetto soda pop. They didn't have soda pop. My mom wouldn't buy soda pop. She'd make Kool-Aid. And on the directions for the little package of Kool-Aid, it says one package makes 2 quarts. Not for my mom. One package made like 2 gallons. And what you know what the most common Kool-Aid is? And don't say red, that's a color. Cherry, yeah. Second is grape. My mom got orange. They cost the same. She just wouldn't get us what everybody else wanted. So she would dilute it down. And then when it got to where it was toward the bottom, she added more water. Now it says you're supposed to put 2 cups of sugar in there. Okay. My mom put 2 tablespoons of sugar because she didn't want us to rot her teeth. She put 2 tablespoons of sugar. You couldn't even taste the sugar. But let's say, now when I got by myself, I said, man, I love Kool-Aid and I'm going to make it the way I want it to. So I dumped almost a half sack of sugar in there. Well, you know what happens when something is super saturated? In other words, it has so much stuff in it, it precipitates out at the bottom. So I poured all the sugar in there and I took that spoon and I whipped it around and guess what? It still settled at the bottom. There's too much sugar. I said, hey, I'm a scientist. I know what to do. I heated it up. When you heated it up, it'll put all that sugar into solution. I outsmarted it. Yeah. But it was so sweet. I'm telling you, one sip and your eyes crossed. Okay. So the heat was a catalyst. The spoon was a catalyst. You got it? Heat's a catalyst for a lot of chemical reactions. Okay, so now you know what a catalyst is. An enzyme is a catalyst in a living organism. Yeah. An enzyme, one more time, because it's a test question. An enzyme is a catalyst in a living organism. Pretty cool, huh? A catalyst lowers the energy of activation. 2 test questions, just gave them to you. Okay, what does the protein do? Cell-to-cell recognition, along with that glycocalyx. Signal transduction. Okay, transduction means you go from one to the other. Sends a signal along. Not all cells, some cells. That's how they communicate, through protein. Okay, now transport. Now we're going to transport things. Remember, selectively permeable? Selectively permeable means things are permeable, but only certain things. Okay? Amino acids can go into one cell, but they can't go into another. How come? Because it's selective for certain things. Does that make sense? Okay. It says influenced by certain factors. Type of protein. That integral protein, not all proteins are the same, Jacko. They're all different. So some will allow certain things in, won't allow certain other things. So the type of transport protein, whether it's an integral or peripheral, doesn't make any difference right now. We're just talking about those proteins, those blue and green ones, make what is selectively permeable of a cell. Phospholipid bilayer doesn't do it. It's the integral proteins. Plasma membrane structure. We're talking about how does it get through. Concentration gradient. Oh boy. Okay. Now there's I rewrote the transport cell membrane transport because the book was too darn confusing. So I want you to go by mine. Okay? I think I made it really simple. You have to have a concentration gradient. That means more on one side, less on another. Okay? Draw a line on your paper right now. Draw a line on your paper, but don't make it a continuous line. Make little tiny spaces. As you're drawing the line, skip, go again, skip, go again. Okay, have you done that? Do it on the other side of your page that you're not doing anything on. You're putting pictures over there or something. Okay, now put 10 X's on one side. Just make an X, 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 X. Like you're gonna play football. Only no O's. Got it? Got it? Now, on the other side, I want you to put O's, put 10 O's. Just circle little O's. Zeros if you want. Okay, now you have 10 X's on one side and 10 O's on the other. You have 2 concentration gradients. X's concentration gradient, O's concentration gradient. How many O's do you have on the other side? 0. How many X's do you have on the other side? 0. So your concentration gradient is on one side or the other, depending if you're talking about X's and O's. Does that make sense? Now, remember those little lines I made gaps I told you to put in there? Remember that? Okay. If that gap is big enough to allow the X's and O's to cross, what will happen? They'll move, huh? How many will move? Well, they'll all move. No, they won't, because then they're going to be all on one side and on the other. You still got the same thing. Half of them will, huh? So you'll have 5 X's on one side, 5 on the other. That's called isotonicity. They're the same. Then 5 O's will move over, and you'll have 5 X's and O's on one side and 5 X's and O's. It's balanced. You don't have a concentration gradient anymore, Jacko. Got it? Now, what about if the O's are too big to get through that little space? It's impermeable. Impermeable, huh? Won't go through. So how are you going to get them in there? You have to put a bigger protein in there, open up that little door, huh? Does that make sense? Concentration gradient. More on one side than another. Ionic charge. If something is charged, positive, you only got positive and negative. If something is charged, that means that protein has to allow it to come through. Okay? And you know, like charges repel. So if that protein has a positive charge, and what you want to come through is positively charged. Let's say you have calcium. Calcium's got a positive charge, or sodium has a positive charge. It won't go through there, Jacko. That means that protein has to have a negative charge, and then allow it to go through. That makes sense? Ionic charge. Lipid solubility. Remember we talked about this one? Okay. When you're on the outside or when you're on the inside of the phospholipid bilayer, you're surrounded by water and you are happy if you're polar. But if you are not polar, that means you're a lipid, you are being repelled. So how do you get into a cell? Well, you catch a ride on a protein. So once you close the car door, you don't know who's in it. So the lipid is sneaking up to the cell. Once it gets to the cell, remember that fatty acid? Hey, that's its cousin. It's going to go right on through, isn't it? Okay. Lipid solubility. Molecular size. I want to tell you about a fat person going through a door, but I know I'll get in trouble for it. So pretend like I didn't say a fat person going through a door. If the door is only 2 feet wide and that person's 3 feet wide, are they ever going to get through that damn door? No. You got to build a bigger door. Molecular size can inhibit it, can it? Okay, I didn't talk about that. Two general types. Now this I have done differently than the textbook because I think I can explain it easier. There's two, it's like 2, 3, 2, 3. Got it? There's two types of transport. Got it? Two types of transport. Active, passive. Just look at the green right now because I'm going to erase the other stuff. Passive and active. Okay. Passive doesn't require any energy. Concentration gradient. I got more on this side. I got less on this side, and I can go through that door right there. See that door right there and the Great Wall? If I can get through there and there's more on this side and less on that side, I'm going to go through that door. Okay, which is your integral protein. Okay? And this is where we hear about diffusion and osmosis. Got it? And I'll talk about those, but you're going to see a blue one up here. It says substances move down their concentration gradient past a membrane. It's called diffusion. Any stuff, any molecules that go from higher to lower concentration gradient is called diffusion. You understand? Okay. Diffusion is a type of passive transport. Got it? Does not require energy. There's only one source of energy in a cell. Only one source of energy in a cell. Guess what it is? ATP. Okay? It's ATP. Passive transport does not require ATP. Wow. Got it? Passive transport, diffusion, osmosis does not require ATP, energy. But the next one does. Active transport. Definition. Requires energy. Substance move against the concentration. Ooh. Remember those X's and O's? What about instead of having 10 X's on one side and 0 on the other? What about if we had 7 on one side and 3 on the other? And we wanted those 3 to go over to join the 7. That's a concentration gradient, baby. It ain't going to happen unless you do what? Push it. Push it. That means you're going to require energy to do it. That's what active transport requires, energy. And it's going against the concentration gradient. Good? Requires ATP. Now, there's 3 questions I'm looking at right there on your exam. 2 types of transport. What are they? Passive and active. Passive, example, diffusion. Active, what is it? Against the concentration gradient, requiring ATP. Passive, move it in the direction of the concentration gradient. Got it? Make sure you know these. Write them and put them down and practice writing them until you got it squared away. Passive transport. There are 2 types of diffusion. Okay. Now this, remember I said it, this varies a little bit from the way your book does it, because I think the book was confusing. So I made it to the way I learned it, and I think it's easier that way. There's 2 types of transport, passive and active. Passive and active. Active requires ATP, goes against the concentration gradient. Passive, direction of the concentration gradient, no energy. Good? Now, passive. 2 types of diffusion. Okay, there's 2 types of diffusion. Now I'm gonna use some I'm going to use some chemistry words for you, and you got to learn them, okay? So don't resist. There are solutes and water, okay? And our body is made up of solutes and water. The movement in and out of a cell of solutes is called dialysis. The movement of water in and out of a cell is called osmosis. You understand? The principles are pretty much the same, but the wording they use for it is different. So this is going to get confusing, but if you listen to me and you take good notes, I'll walk you through this. Okay, diffusion, there are two types. What are they? I just told you. Pause it. If you wrote this down, you've got it in front of you. If you're just listening to this with headphones on and going, wow, this is pretty cool, you're wasting your time. You should have written down diffusion is, and then make a split. There's two types. So you have diffusion in a box, okay? Diffusion in a box. You wrote diffusion, put a rectangle around it, and then put two little legs on it. One leg says dialysis, the other one says osmosis, doesn't it? Osmosis is movement of water only, okay? You put a book on your head and you want to gain all that knowledge. You want all that knowledge to go through that book and into your head. First thing ain't going to happen. I tried it. The second thing is that's not osmosis, Jacko. That would be dialysis, if anything. Okay, there ain't no water coming out of there. Okay, and then dialysis, movement of solutes. Osmosis, movement of water. End of subject. So remember, if we made that little flow chart, you had dialysis here. No. You had diffusion here, and you had dialysis and osmosis. Osmosis is a dead end. It's only movement of water. Dialysis, there's three types. Okay, simple, and they call it simple diffusion. They don't call it simple dialysis. I still not been able to wrap my head around this. Okay, if I wrote a textbook, I would call it simple dialysis, because you got to remember it's just a type of diffusion is dialysis. So I put in parentheses dialysis. So simple movement of solutes is called simple diffusion. Got it? That means it can move through the phospholipid bilayer. Remember those purple balls? It can go right through there. Oxygen and carbon dioxide goes right through there. Water goes through there, okay? Because it's real simple. Concentration gradient. More on one side, not as much on the other side, can go right through. Got it? The next one is called facilitated diffusion or dialysis. Now on the test, you know I'm going to ask you these. On the test, I'm going to put it down the way it's supposed to be, diffusion, okay? Okay, facilitated, what does that mean? What does the word facilitated mean? Okay, to facilitate something. It means help it, huh? I facilitate it, I help it. Facilitated diffusion, I help it get inside. Yeah, facilitated diffusion. Guess what that's gonna be? A protein. And there's two types of proteins, integral and peripheral. Oh my gosh. Sometimes we call those carrier proteins. Sometimes we call them transmembrane proteins. Remember that? So we're going to help it by using that door, okay? And then bulk filtration. That's the third type, bulk filtration. That's moving a lot of the same thing. Passive transport, osmosis. Okay, now we're going to give you some definitions. This is the way you're going to see it on the exam. What is osmosis? Movement of water, okay? Now, instead of using the word higher concentration, lower concentration, which we call concentration gradient, which it still is, we use osmotic potential. So a science word, osmotic potential. It means concentration gradient. Got it? So diffusion of water, same principle, simple diffusion, osmotic pressure, osmotic potential, pressure, same thing. So what it's saying is, I got more water over here, I got less water over here. We don't call it a concentration gradient, we call it osmotic pressure. Got it? Still the same thing. I got more here, less here, it's going to go in this direction. So until it balances out. But we don't use concentration gradient. That's no. That's only for solutes. That's only for dialysis. We call it osmotic pressure. Good? Okay, simple diffusion. Now, simple diffusion. That was osmosis. Remember? Remember? Square box, diffusion, osmosis, dialysis. Osmosis, one type, water. Concentration gradient is not called a concentration gradient. It's called osmotic pressure or potential. Now we're going to do the dialysis. Now we're going to move everything else. I don't care if it's an amino acid, a carbohydrate, a monosaccharide, a booger. It doesn't make any difference. That is dialysis. Good? Examples. O2, CO2, in and out of the lungs. Higher concentration, into the blood, lower concentration. Got it? When you breathe in oxygen, what makes it want to go into the blood? Concentration gradient, because how much oxygen's in the blood at that particular point in your lungs? Not very much. I want to say none, but there is a little bit. But how much did you just breathe in? 100%. So you have a concentration gradient. Does it require a protein? No. It goes right through the phospholipid bilayer, therefore it's simple diffusion. You get it? Okay, move through the phospholipid bilayer. Next one, facilitated. Remember, you're going to hear two things: carrier protein, or integral protein. For large and or polar molecules. Transport protein. I just said two of them and it wasn't one of the two. Okay, carrier protein is a transport protein, which is an integral protein, which is a transmembrane protein. They're all the same thing, man. Okay, so in here I put carrier. That's the way I learned it. Anyway, a transport protein, carrier protein, integral protein, same thing. Okay, four. Gotta go past that membrane. And then bulk filtration, large amounts of substances across plasma membrane. Good, good. Active transport, movement of substances past the membrane against the concentration gradient. What's missing? What's missing? You know. What should you look for? You better look for it on an exam, because here it comes. ATP. Energy. Active transport. Now, we're not talking about water. Water's out of the thing. This is substrates. I mean, a solute, sorry. It's solutes. Okay? Could involve bulk transport. Doesn't make any difference. You've got to have a carrier molecule or a transmembrane molecule or a transport molecule. That's your integral protein. And you're going to go against the concentration gradient. You're going to make sure all the X's are on one side and all the O's on the other. Now we see different types. Exocytosis and endocytosis. Come on, think about it. Exo sounds like what? Outside. Endo sounds like what? Endo. Come on, enter. Inside. So when we move things into a cell, it's called endocytosis. When we move things out of a cell, it's called exocytosis. Okay? Remember when I showed you that prototypical cell and I said there's the Golgi body that takes a vesicle, and there was one of them that was dumping stuff outside. You saw little bits of stuff outside. I said they call that a secretory vesicle. Guess what? What you were looking at? Exocytosis. Now, if those little bits and stuff were coming inside and you were forming that vesicle, it would be called endocytosis. Get it? Requires a transport protein, carrier protein, transmembrane protein, all those things. Remember all the names. Okay, it requires that. You gotta have it. It does not ever go through phospholipid bilayer. Example, ion pumps. Your nervous system works on pumping sodium and potassium, pumps it. It's like you can pump water, okay? But in your body, your nervous system, you have ion pumps. And that's an example of active transport. That's what runs it. Electricity runs the pump that pumps water, okay? In your body, the pump is run by ATP. Example, sodium potassium pump, okay? Okay, and I might ask you, sodium potassium pump is an example of what? Active transport, okay? Okay, sodium, potassium, that's how you abbreviate it. Sodium plus, potassium plus, opposite directions, one another, concentration gradients, okay? That's your X's and O's, okay? And then bulk transport. Bulk is just what it sounds like, a whole bunch of it. Try to get a bunch of it in, a bunch of it out. There are two types, exocytosis. You make up a whole bunch of protein, you want to offload it outside the cell. Okay, that could be anything. It could be a digestive enzyme. Okay. It may be, let's just use that, a digestive enzyme. You make a whole bunch of, the cell makes a whole bunch of it, and you want to dump it off in the stomach. What are you going to do? Wish it out there? No, you're going to get rid of it by bulk transport. Example is exocytosis. What about if you have something that you want to bring into the cell? A bulk inside the cell. That's going to be endocytosis. Like what? Bacteria, virus. You want to bring it inside the cell so you can decompose it? How are you going to get in? Endocytosis. Got it? Exo, endo. I'm going to ask you that on an exam. Bulk transport, exocytosis. What is it? Okay. Remember in the Golgi body, remember the phospholipid bilayer? Remember that? Phospholipid bilayer? Remember Golgi apparatus? And we saw those little green circles that were made, and they were kind of moving over towards the cell membrane. Remember that? Remember that? Okay, those are vesicles. And if they're taking something outside the cells, they're called secreting secretory vesicles. And it means they're just a suitcase full of a protein. It's going to dump it outside. Process of dumping it outside called exocytosis. Do you have to have a carrier molecule? Remember integral protein, the green and the blue? Do you have to have one of those? Yep. Yep, you do. You're saying, but I have concentration gradient. I have a whole bunch on this side. I don't have any on the other side. Yeah, but how are you going to get your fat across there? You got to have a carrier molecule, okay? Because it won't go through the phospholipid bilayer. There you go. Endocytosis, opposite of exo, bringing it in. Good. Once it's in, we put them in vesicles. So whatever's coming into the cell by bulk transport, we wrap it in a package, a suitcase. The suitcase either goes to the outside or suitcase comes on the inside. Still got to put it in a suitcase. You can't just kind of hang out. Okay. Exocytosis. I got the steps in here. You can read the steps. And I'm going to have a picture on the right-hand side. Okay, it's kind of a dumb picture, but it's a picture that you're going to see in your textbook. But I showed the steps. 1, 2, 3, 4. Okay, and here's the picture. This is extracellular fluid. So we're going to go, this is exocytosis. So this is the inside of the cell. You made this in the Golgi, and this is all the protein. So what you're going to do is, there's your carrier molecules. So what you're going to do is you're going to move it up to here. They call this evaginating. The phospholipid bilayer evaginated, pops it out, and then this vesicle becomes part of the plasma membrane. Did you get that? Okay. Read up on that. You're going to need to know because I'm going to ask you a test question. But I can't just sit there and go over it and say it 20 times. It's not going to make any difference. You have to read it, turn off the computer, and memorize it. 3 forms of endocytosis. Exocytosis, one type. Exocytosis, get out of here. Okay? 3 types of endocytosis. Yes, test question. Here we go. Phagocytosis. Now take it apart, because here's what we're going to do it. Cytosis, cell, phago, to eat. Eat a cell. Okay? So what that means is you're going to invaginate. Your phospholipid bilayer is here. What you're going to do is you're going to start to cave in. And then you're going to wrap right around it, and you're going to make a vesicle out of it. Okay? And you're going to eat it. Phagocytosis. The other one, pinocytosis. Whoops. Here's a picture. Okay, so this is coming in. You see how it's invaginating? So this should be like 3 or 4 different pictures, but they're saying this comes around, it'll wrap around itself, it'll form a vesicle with the goodies inside. That's cell eating, phagocytosis. I'm just going to ask you on the exam what the definition is. Okay, cell drinking. I always in class I always use this as I said, you know how a fly eats? F-L-Y, fly eats. They actually take digestive enzymes, spit it out of their mouth on whatever they want to eat. Usually it's poop, okay? They want to spit it out, and that will dissolve it, and then they lick it. They drink it. Do you know that? True, okay? It's called cell drinking. Pinocytosis is cell drinking. Uptake of extracellular fluid, okay? And this picture doesn't show it, okay? It doesn't show it. What they did is they said, I've already put the digestive enzymes out here. I'm going to package them up and bring them inside as pieces. Just to eat the whole darn thing. Okay, the other one is the third type is receptor mediated. Now, you know I'm probably going to give you an ABCD on this one. Receptor mediated means you got a specific arm sticking out there. Okay? You got an arm sticking out there. So this arm is attached to my body. My body is the phospholipid. Okay, The receptor mediated, guess what that is? Peripheral protein. It's out there saying, come out here, gotcha, and I'll bring it in. Good. Two-step process. Okay, blah, blah, blah. Read that. Okay, I'm not gonna read it to you. Cytoplasm. Okay, I'm gonna take a breath. I'm gonna take a little quick little break. Okay, you're not gonna know because I'm just gonna shut the darn thing. I'm gonna pause this thing and I'm gonna come right back. All right, we're back. I'm having some barbecued pork. Because this, remember I said we were going to take it in 3 sections when we talked about the cell. We're at the phospholipid bilayer, then we're going to look at the cytoplasm, and then one portion of the cytoplasm nucleus we're going to look at separately. Remember that? Okay, we just did the phospholipid bilayer. That took 33 slides to get that down because it's not an easy walk in the park. Okay? Now we're going to go to the second thing. This is pretty straightforward. I would imagine when you were in middle school or something like that, you had a project that you had to go get a styrofoam ball, cut it in half, and put little things in there that represented organelles. Remember that? Okay. Okay, and you probably told your mom on a Sunday when it was, and you had to go down to Michael's or Joanna's to get it, huh? Okay. Times have changed. We're going to be talking about those things inside the cell. More than likely, the teacher had you get up and you took one organelle and you talked about it, what its name was, what it did in the cell, and that kind of junk. If not, you didn't have a very good science teacher. Okay, it says anything inside the phospholipid bilayer, anything in it. Let me take that back. The jelly-like fluid inside the phospholipid bilayer is called cytosol. If you add the organelles with it, it's called cytoplasm. That's what we're talking about. We're talking about the cytoplasm. Got it? So the way to do it is, we're going to talk about first, I'm going to have a little hit of Crystal Geyser. What we're going to talk about first, we're talking about what the cytosol is. What's it made of? Okay? And then we'll go to the organelles, and we'll go through each one of those. And what I'll do is I'll give you a name. We'll define it. We'll talk about what it is. Okay? And then I'll show you a picture of what they look like. And I'll take that picture right out of the prototypical cell. Got it? Okay, so that's the way we're going to attack it. And we'll go through each one of the organelles, talk about the general things they do. Some of them you're going to know, some of them you're not going to know. But hopefully, by the time of the lecture exam and the lab exam, you do know what they do. Okay, because you know I'm going to ask you. Okay? Something happened here, Jacko. Let me get this back. See, we'll go like this. Oh, there we go. Okay. Cytosol. Jelly-like fluid. We already talked about it. What's it made out of? Okay, then we have inclusions. Inclusion, remember I said there's those other things. Things that kind of added in. You're going to see melanin. It gives you skin color. Keratin gives you skin color. Okay, we'll talk about those. And then the organelles. Yeah, and the organelles. Okay, so here we go. Cytosol. What's it made out of? Viscous. Viscous means how thick something is. Jelly-like fluid containing many different dissolved substances, such as ions, calcium, sodium, chlorine, chloride. All those are ions. Anything that has a charge on it is an ion, whether it's negative or positive, it doesn't make any difference. Nutrients. What's nutrients for the cell? Monosaccharides, amino acids, fatty acids, nucleotides, those are all nutrients for the cell. And that's floating around in the cytoplasm, the cytosol. Proteins. Now, I got this one kind of screwed up because I put proteins, carbohydrates, and I put lipids, and then I put, oh, I didn't get it screwed up, I fixed it. Look at those, look at those. 1, 2, 3. There's 4 macromolecules. The only one that's missing here is nucleic acids. Good? Okay, anyway, that's what's in the cytosol, all that stuff. Inclusions. Okay, let's talk about inclusions for a minute. Okay, one of the ones we're going to talk about is melanin. Melanin is a pigment. The cells that make melanin are called melanocytes, and they're mixed in with your skin cells. I know it's impolite to chew, but it's going to take me 3.5 hours, 4 hours to go through this. I would be famished. Okay, so in my skin, I have very little melanocytes. I have friends that have a lot of melanocytes. We know them as black people. They know me as white people. We have Hispanics that are kind of like in between because they have a tan. Okay, that is due to the number of genetically, what we are composed of genetically as far as how many melanocytes we have. You've seen people with really, really, really dark skin. I have 0. Weird, weird. Okay, skin looks like paste. Looks like Elmer's glue. If we were in person, I could make it more funny, but we're going to get right on with it. That's going to be some of the stuff in inclusions. Melanin. Melanin isn't the inclusion. Melanin is the pigment that is found, and we call that an inclusion. Okay? Sometimes I've seen some books that write it down as brown, and it's not. Melanin is black. Okay? The less black you have, the more brown you have. The less brown you have, the more white you have. Okay. Glycogen. Now, glycogen is a whole bunch of glucose molecules hooked together. Remember that pearl necklace we talked about? When you have each one of those pearls is a monosaccharide, and the monosaccharide is glucose, and you put a bunch of them together, you get what's called glycogen. We do that in our body. Diabetics have too much glucose in their blood, which means something's got to be pushed out. So they offload it in their urine, but they'll have problems with their metabolism and stuff because they have too much glucose, and that means they don't have enough of something else. Okay? And normally, insulin takes glucose out of your blood and stores it into cells, muscle cells, liver cells, and eventually adipocytes for lipids. Got it? Then we have another enzyme, another hormone, that's insulin is a hormone. We have another hormone called glycagon that takes glycogen out of those cells and puts it into the blood when we don't have enough glucose. Okay, so anyway, glycogen is a type of inclusion. Carotene. Carotene is an orange-yellow pigment. You may have heard they talk about Asians as the yellow brothers, or what else do we hear derogatory? Yellow brothers and sisters, the yellow man. Okay, when we were fighting them during Vietnam and stuff, it was the yellow man. And they go, what the hell is a yellow man? Okay. It just means that instead of having melanocytes producing melanin, the pigment, they have keratinocytes producing keratin. It gives them that little bit different color. Okay? Interesting, huh? Organelles. Okay, now we knew that did the inclusions. We did cytosol, and we did the inclusions in cytosol. And now we're going to do organelles. Okay? Means little organs. I'm not going to ask you that. Many types form different functions. Obviously, they form different functions, or you'd only have one type of organelle. I mean, duh. Okay. So, and we can also figure this, because this is kind of a duh too. If we have a cell that requires a lot of energy to do its job, what organelle do you think it's going to be full of? Mitochondria. Yep. If we have a cell that its major job is to store, what's it going to be full of? It's going to be full of vesicles. Yes, which we call vacuoles when they're bigger. How come? Because they don't need a lot of mitochondria. That makes sense? Okay. Division of labor. This is not vodka, it's water. I don't drink anymore, or any less. That's money on. Okay, division of labor. Your cell is set up so every organelle has its own job and only a single job. It isn't going to say, oh, hey, man, you want to take a rest? I'll go ahead and take over your job. Okay, and I go into this little scenario about division of labor. I'm going to ask you this question, and do not wait for the answer. Pause and think. Where did we see in America the first division of labor? And I'm not talking about your mom and dad telling you to get outside and rake the lawn. Where did we see division of labor? Okay, it was something that as soon as I tell you, I'm going to go, well, duh. Okay. You didn't do what I asked you. You didn't think about it. Okay. The way they used to make cars is you came to work, and there was a pile of parts out there, and you built a car. From nuts and bolts and tires and wheels, you did it. You. When you were done, you stamped it with your name. And then you went and got to a different pile. You know where I'm going with this. This guy named Henry Ford came around and he says, that's a waste of time. We can speed that up and therefore cheapen the automobile by having an assembly line. Now, mind you, for those people that are like me, would go bored in a 2 days, but you become extremely efficient. Your job for 40 years is to put steering wheels on, okay? Nobody can put on a steering wheel like you. You could put on a steering wheel with your eyes shut. Another guy puts on tires, okay? And you do that for your whole life. But how fast can you put on a tire? Like in a nanosecond? I met a guy that came into my class and he worked at in the assembly line over in over off the Bay Area. They were making some cars over there. I don't remember what he told me. And he was in charge of putting in brake lines. That means the master cylinder and all the lines go into the wheels. And I said, this is going to scare you. I said, how long does it take you to put in, take it out of the box, or wherever it's stacked up, the cylinder, the master cylinder, and then run all of those in and anchor them on the frame, go into each wheel? He says 15 to 20 seconds. I went, I'm scared when I press on the brake. And he says, what you do is you walk as the car goes down the assembly line and you're putting this stuff on. And he says, I can get it done in 15 seconds, go grab another one, be ready right when it's leaving my buddy that's next to me that's putting on the wheels. Okay, you see what Henry Ford did on the assembly line? This division of labor. Okay, I'll take you one other example. This is what I call soft skills. This ain't gonna be on the exam. If you don't like it, fast forward. Fast food, which we see today, and we're so used to it, it isn't funny. But they used to have what was called a grill. You went into stores, like let's say you went into Sears. I remember Sears had one, and Penny's and Macy's and Dillard's and those things. And they had a what they call a grill portion where somebody was working there, and you went in, and that's how a cafe was. And the cafes still are like that. Like when you go into a sit-down restaurant, right? You choose from the menu what you want. The person comes to you. They say, what would you like? And you say, well, I'll have a burger, fries, and a milkshake. And okay, what do you want on your burger and that kind of stuff? Then they went into the back and made it. And about a half hour later, you got it. Okay. That very efficient? No. She's got to take the order. He has to take the order, go make it, bring it out to you, then bring you the bill and have to make change and then do the cleanup. That is not smart. So what's fast food? Was the first one to go into assembly line. Come on. It wasn't Taco Bell. The old double arches. McDonald's came out with an assembly line. Now, when you go to In-N-Out Burger, you get to see him doing it. You got one guy over there peeling potatoes, making French fries, one making the sodas, one doing the burgers, and one taking your money, and the other one handing you the food, right? That's division of labor. Do you know how efficient that is? You have learned to live with that. You don't get your food within 5 minutes, you're pissed off. Okay? Anyway, division of labor. Your cells do the same thing. Same thing. It's an assembly line. Okay? All right. Thought you'd find that interesting. Division of labor you'll see on your exam. And this is a kind of a no-brainer. The type and number of organelles within a cell is a reflection of the cell's function. If you're a kidney cell, you are full of ATP. You're full of mitochondria that's making ATP because you are going through active transport like there is no tomorrow. Got it? Because you're constantly doing active transport, putting things in, putting all those X's on one side and all the O's on the other, constant flow, which requires ATP. So you're gonna be full of mitochondria. You think you're gonna have very many vacuoles? Nope. You think you're stored much? Nope. Make sense? That's what that bullet says. Okay, organelles. As scientists, can we break those organelles into categories? The answer is yes. Another hit. Crystal geyser. I like the way this thing fades out. It looks like I'm putting something in my mouth and nothing's there. Organelles can be classified two different ways. Those that have a membrane and those that do not have a membrane. Got it? So we call those membrane bound and non-membrane bound. Got it? So in our exploration of organelles, what we're going to do is we're going to take membrane bound. We're going to go through them all. Okay? The major ones. And I'll tell you what they are by definition. What do they do by definition? I'll show you what they look like in that prototypical cell. Got it? What their function is. Okay. And non-membrane, then we'll do non-membrane bound, and then we'll do cytoskeleton, and then we'll do the nucleus. Okay. Okay, here we go. Membrane bound. Example, endoplasmic reticulum. When you were in school, if you took a science class, you probably called this ER. Okay? That's great. You knew it as ER. And anybody can spell ER, right? But endoplasmic reticulum, you couldn't spell. Now you got to spell it because I don't want you to write ER. If you write ER in the lab test, you'll get it wrong. I want you to write endoplasmic reticulum. Just like DNA. I want you to write deoxyribonucleic acid. You're in college. You're going to learn these things. Good? So ER, there's two types, smooth and rough. Golgi apparatus. If you see it as Golgi apparatus, Golgi body, Golgi, anything Golgi, it's still the same thing. Okay? Modification, packaging, and secretion. I'm going to do that for you in just a minute. Lysosomes. On our prototypical cell, it's a round ball and it's yellow. And it's for intracellular digestion. It's like your stomach of your cell. Lysosomes. I mentioned it once before when we saw the Golgi body. And I said some of those suitcases go to take stuff outside, some stay inside, and we call them lysosomes. I showed you on the prototypical cell what it looked like. You don't remember that because you weren't paying attention. Okay. Peroxisomes. Okay, for you people that partake in drugs and alcohol, which I hope there's zero out there, when you detoxify your body. You don't detoxify your body, you detoxify your cells. And the little organelle that's out there working to try and straighten your goddamn thing out on the next day when you feel so terrible is the peroxisome. Okay? And they take hydrogen peroxide, catalyze it, and turn it into water. Okay? Okay? And poisons. That's one thing, and they do poisons. Mitochondria, the powerhouse of the cell, ATP. Here we go. Endoplasmic reticulum. What's that say over there? I can't see it. My picture's blocking it, so I don't know if it's doing yours. We'll take a peek. Oh, ER. Okay, endoplasmic reticulum. This is how it's going to be on the exam. A series of tubes and channels for transport of proteins and other substances. Write that down. It's going to be on the exam, and it's going to exactly be written like that. But that barbecued pork's good stuff, huh? Series of tubes and channels for transport of proteins and other substances. Wow. 2 types. What did I say they were? I mentioned it a minute ago. You should have written it down. If you didn't, you said, oh, I didn't have time to write it down. Man, you got a pause key. Okay. For those of you that did, here we go. Smooth. Now, I'm not going to write it S-E-R. It's smooth endoplasmic reticulum and rough endoplasmic reticulum. Easy, easy, easy way to remember it. Rough has ribosomes attached. Smooth does not. Okay. Still a series of tubes and channels, but they're connected. So it might be rough here and smooth here. Oh, my finger disappeared. It might be rough here and smooth here, and then rough again. Okay. The only thing that makes it rough is it has ribosomes attached. Smooth does not. Okay. And there's going to be a function of those differently. Here we go. Endoplasmic reticulum. This is rough. Do you see it? You see right here? See all the little dots? Those are ribosomes. Do you see no dots? Okay, that's smooth. Here's what it looked like in a transmitting electron microscope. Remember that? Transmitting electron microscope. See all the little dots? Okay. So this is rough ER. You don't see any smooth ER. This is rough ER. And when a ribosome is attached to the ER, it's called attached. Duh. Okay. When it's attached to the ER, it's called attached ribosome, attached ribosome, or fixed ribosome. When it's not, when it's out here like this, okay, it's called free. It's free of the ER. Free ribosome. Got it? Now, you see what it says under a detoxification? This is for smooth ER. This isn't for rough ER, smooth ER. It says smooth ER. What does it say? Detoxifies both drugs and alcohol. Smooth ER. So you got this whole branch of smooth ER in there. Remember what I said peroxisomes do? Smooth ER does it too. Okay. And rough ER just transports proteins for exocytosis. Okay. Smooth ER lacks ribosomes, duh. Continuous with rough ER, I just told you that. Functions include synthesis and transport and storage of lipids, including steroid hormones. Listen, synthesis, transport, and storage of lipids, metabolism of carbohydrates, break down carbohydrates into sugars, monosaccharides, and detoxification of drugs, alcohol, and poisons. You know alcohol is a poison. You know that? Okay. So as long as you got smooth ER and peroxisomes, I guess you're okay. Okay. Don't do that. It's not good for you. Rough ER. Attachment of ribosomes. You see what it says that right there? Attachment, ribosomes. The ribosomes are called attached or fixed, and the ER is called rough ER. Don't worry, I'm not going to die. I got to get through this lecture. Oh, I know you felt bad for me, huh? No, you didn't. Okay. I tried eating that darn fork too fast. Okay. A ribosome that's attached is called fixed or attached, and it's attached to ER and it's rough ER. When it's smooth ER, it doesn't have any ribosomes attached. Ribosomes that are not attached, has nothing to do with smooth ER, are called free. Free, as in like you like to get stuff free. Okay. Big thing. Ribosomes synthesize proteins. What does synthesize mean? Turn off your computer, tell me what it means. Okay, you should have come back with synthesize means to make. Yes, all those proteins, whether it's hair, whether it's the proteins that go in your skin cells, whether it's enzymes, whether it's hormones, whether it's cytoskeleton, whether it's antibodies, those are all proteins, and they're all made on ribosomes. The only place they're made is on ribosomes. You get it? It says rough ER transports proteins. That's how it moves in the cell. Once they're made, they're put into a tube, and that tube goes to the Golgi. And then it's secreted. It goes to the Golgi, it's put into a suitcase. Remember that? A secretory vesicle, secretory suitcase, and it's secreted. Exocytosis. It says it's incorporated into the plasma membrane. Now, remember that? It evaginates and makes a thing, dumps it off, becomes part of the phospholipid bilayer. I showed you a picture of that. Golgi apparatus. Here's the way you're going to see it. Look at it. Right here. Right here. Just a minute. Right there. Modification, packaging, and secretion of proteins and other substances. Don't put vesicles there. Don't put vesicles there. Modification, packaging, and secretion of proteins and other substances. You got it? Okay. Now, what does it do? Puts them in a suitcase. What's a suitcase called? A vesicle. Got it? Remember that? You've seen pictures of it. Okay, this is just going over it again. And it is, remember how it had that picture and it evaginated, then it pouched out. Okay, that's what that's showing you does. That's what it is. It bulges out, pinches off, gives rise to small transport secretory vesicles. Okay. Remember where the gold was in the prototypical cell and it has like a bunch of little squish things? That was actually a tube or a channel cut open so you could see inside. And remember there was a series of them that come off the nucleus, and then it went to a green thing, and the green thing was a Golgi apparatus, Golgi body. Yeah, that okay. The side closest to the nucleus that is receiving the proteins from the ER. Once again, the side that is closest to the nucleus that is receiving the proteins from the rough ER is called the cis phase. That's what's in blue, cis, C-I-S. Okay, that's a cis phase. I'm sorry, if I said phase, I meant space, this phase. And then when it goes through the Golgi, it's all A's are put together, all B's are put together, all C's are put together, all D's are put together. They don't put A's and B's and C's together. You separate them. When that's done, it goes out the other side heading for the phospholipid bilayer, and that's called the trans phase. Got it? Cis phase, entering, trans, going away. Shipping, receiving. You get it? I'm going to ask you on an exam. Simple as that, memorize, but next. Golgi, picture. Okay, see this side? What they don't show you is the nucleus. This is what I was trying to describe to you. This is the cis phase. This is the trans phase. It comes to here and it's a mixture. A, B, C's, D's, E's, F. As it winds through this warehouse, it packages them up. So all A's are there, all B's are there, C's are there, D's are there, so on and so forth. Now, these are going for exocytosis. See it coming out? These are turning and becoming, just a minute. Mine went blank. Intracellular digestion. Lysosomes. This is a lysosome. It's yellow. Okay, it's not yellow in the cell, but they made it yellow here. And these are secretory vesicles. See the little stuff coming out? Those are proteins. Wow, pretty cool. Okay. Now here's a step 1, 2, 3, 4, 5. Kind of like we did it before. I said, how does this work? Okay, proteins come into the Golgi from the ER. It comes in by way of the cis face. Okay, and there it's packaged up and modified. Like things are going into one, and unlike go into another. So A's go with A's, B's go with B's, C's go with C's. Those are different types of proteins. Got it? If you have to add something to it to make it active, it's done there also. That's why the definition said modification, packaging, and secretion of proteins and other substances. Got it? Modification, you add stuff to it. Okay. They're put in transport vesicles. The transports that go outside the cell are called secretory vesicles. The ones that stay inside are called lysosomes. Okay? And it tells how it does. Pinching off, remember the picture? Pinches off, modified, packaged in secretory vesicles. They're going outside, stay inside their lysosomes. See, there's the lysosome. Are we good? That's straightforward. I'm not going to spend a lot of time because you can read that. It's an insult for me to read it to you. Okay? You can read it. I won't do that to you. Golgi apparatus picture. Okay? Cis face, receiving. Trans face, going out. This is going out. Exocytosis. We talked about that. Exocytosis. Secretory. Lysosome. Stays inside the stomach. Intracellular digestion, which we're going to come up to pretty darn quick. There they are. Can't get any quicker than that. Lysosomes. Definition. Intracellular digestion. Whoops. It says it's generated by the Golgi apparatus. Intracellular digestion. Okay. Now don't look at the blue right now. I want you to write intracellular digestion. Anything that comes into the cell, lysosome has the enzymes to break it down, which are proteins. The enzymes are protein, the protein enzymes, and they break down anything that comes inside the cell. What else it does is it breaks down a cell. Let's say your cell is so jacked up, you have just abused that cell, and it says, hey, I cannot fix myself. I'm better off to use me for parts. Break me down and use me for parts. When you break down a cell, you eat a cell, it's called phagy. When you do it automatically, it's autophagy. You automatically break it down. Wow. What triggers that? Well, mitochondria and lysosomes. They trigger autophagy. And then once the cell is all decomposed back into its nuts and bolts, then we go to the next thing called autolysis. Lysis is to break open automatically. So what happens is a cell that's so jacked up, you can't just break it open because there's parts in it that's going to get all mixed up with other cells. You're going to get DNA and chromosomes and genes and all that stuff going in there. You're going to get toxins in there because the peroxisomes are gone. So what you got to do is you got to neutralize everything into it. So think of it as a car. You take the car into a garage. When you come back, the car is nothing but a stack of bolts. You're going, what happened to the car? Well, we autophagied it. We broke it down. And then you open up the garage door and you take out all the stuff, and there's nothing in there that can hurt you anymore. That's called autolysis. You get it? So when a cell dies, it dies by autophagy. We decompose it on the inside with the organelles. Main one is lysosomes. And then it breaks open because the phospholipid breaks open, and it's called autolysis when it does that. Did you get that? Okay, here we go. Peroxisomes, yep. You remember those guys, huh? Everybody always remembers those because we talk about drugs and alcohol. Yeah. Anyway, we're going to learn a little bit more too. Okay, they also come off of the Golgi, kind of like the lysosomes did. When the lysosomes have one job, peroxisomes have a different job. They use oxygen and catalase. Now, whenever you see an ASE, it means it is an enzyme. Okay, so the cata means to break down enzyme. It's an enzyme. There's a generalized enzyme that breaks it down. Okay. Here's a good one. DNA. You know what the catalyst is that breaks down DNA? Deoxyribonuclease. RNA, what breaks it down? Ribonuclease. You get it? You get it? Okay. That's how we ended up doing it. It's pretty simple. Anyway, you use oxygen and catalase, a generalized breaker downer enzyme, and it detoxifies toxins and poisons taken into the cell. Pretty cool, huh? Mitochondria. Ooh, look at this. Look at this. We got a new a green thing out here. Double membrane bound. Remember I said there was membrane bound and non-membrane bound. What I didn't tell you is there are certain things that are double membrane bound. Can you think of another one besides a mitochondria? Because the mitochondria is looking right at you. That's a no-brainer. What else is double membrane bound? How about the phospholipid bilayer? Hello, isn't that two membranes? So the plasma membrane is double membrane. Does that make sense? The other one's the nucleus. Mitochondria, nucleus, phospholipid bilayer. Got it? Plants have another one called a plastid. We'll get into that. Okay. Now, we know that mitochondria make ATP. And you want that little mitochondria. Remember that? Oh, I ate them all. Sorry. Remember the little hot tamale? If the hot tamale is hollow and ATP is made inside of mitochondria, and you only have the surface of the mitochondria as a place to make ATP. So what do you want to do if you want to increase surface area? You make little W's inside. Yes? Yes? And we call that a matrix. So think of a honeycomb or a maze. Okay? The walls in the maze or the honeycomb, that is called a matrix. And on that wall and maze is a thing called crista or cristae. And that is where ATP is made. ATP is made on the crista or cristae on a matrix. So whenever you want to increase surface area, I'm going to tell you this several times today. I mean, in the course of this semester. When you want to increase surface area, you're going to have to make some W's inside. You have to make a matrix. Okay? So your intestine, do you want to have a lot of surface area so digested food bump into it and it gets absorbed, or do you want it a straight tube? I hope you said you want it so you have more surface area. So we don't call that a matrix, we call it villi. Okay, still a matrix, but we call it villi. In the stomach, we have, so your stomach can stretch, we call that rugae. But it provides for more surface area. You getting it? The more surface area in a mitochondria is called a matrix, and where ATP is made is on crista or cristae. Got it? Isn't that fun? Okay. Function is to produce ATP on the crista or cristae. Got it? Cells that require more energy have more mitochondria. Now, I hope that didn't, you go, wow, that's cool. I didn't know that. That should be kind of a no-brainer. I gave you an example of it with the kidney, didn't I? Okay, mitochondria picture. Here's our hot tamale. Now, what this is you're taking one of this is a little matrix, and you're pulling it out and you're seeing the crista or cristae on it. Got it? And here's what it looks like at a transmitting electron microscope at 50,000. Let's say 50,000 or say 80,000. I think it says 80,000. Got it? Okay, so they really exist. We're not making it up. Okay, now we did the membrane bound. Now there's more, but those are the main ones. Those are the main guys. Okay, now we're going to do the non-membrane bound. So obviously you're going to see cytoskeleton, you're going to see ribosomes, that kind of stuff. Okay, all right. It says they're in direct control of the cytosol. Oh, duh. If they don't have a membrane, they're in direct contact with the cytosol. Got it. Examples, ribosomes. How many types of ribosomes are there? Come on, come on. Pause it, think, go back, look through your notes. Okay, now you've just come back, you looked at your notes, and you said, there's two, fixed or attached and free. Very good. Cytoskeleton. Remember those little lines? I said we have a skeleton. It gives us shape, and ours is made out of bone. What's the skeleton in the cell made out of? And your answer was protein. Very good. And when we saw the phospholipid bilayer, remember the purple balls and the little yellow legs? And we saw those little wavy lines, and I said, that is a protein collectively called cytoskeleton. Guess what else? Remember the cilia and the flagellum and the microvilli that were on the bottom right-hand side of the prototypical cell? And I talked about what they do. Remember that? Remember that? Guess what? Cytoskeleton made out of protein. Centrioles and centrosomes. Remember when I showed you the prototypical cell and there was that funny looking thing that looked like a gear and it was green and it was at 90 degrees and there was another one? Okay, that is a centrosome. And it's sitting out there, and when a cell divides, they split apart and they move to opposite ends, and they're like an anchor. When they split apart, they're called centrioles. When they're together, they're centrosomes. I will ask you that on an exam. Okay? Cilia and flagellum. Remember, they move a cell unless the cell is attached, and then they move things by them. Flagella, we only have one. Okay, we only have one flagella, and that is a sperm cell. Okay? And that makes that thing whip around like it's going crazy, like it's having a spasmo attack. Cilia, we find that. You saw a picture of it, didn't you, in the respiratory tract? Okay? Microvilli are the really, really short ones. They don't make any movement. They slow food coming down the digestive tract so it can be absorbed. Remember me telling you that? Doesn't cause movement. Cilia flagellum cause movement. Microvilli do not cause movement. Good? Good? They're only found on the digestive tract, and they're to slow the food down. In your book, it'll say increases surface area. Well, yeah, it does increase the surface area because it makes it bigger. But the function is to slow it down. It's like sliding your feet on real smooth concrete and sliding your feet on sandpaper. Can you slide your feet on sandpaper very good? Please say no, okay? Because that grit, which is like the microvilli, slows it down, okay? All right, ribosomes. This is a big thing. It's not a big thing, but they make it a big thing. There's a large and a small subunit. There's like a pot with a lid. Got it? A pot with a lid. And your RNA that's coming out with that code that came from the DNA, the pot lifts up and it goes and slides through it and it's red. Got it? Okay. Responsible for protein synthesis, we already talked about. There are free ones, they're unattached, and then there's fixed or attached. Got it? The attached ones make proteins to go for exocytosis, and the free ones make it to stay inside, become a lysosome or peroxisome. Picture. Okay, there's the lid. Okay, there's the small, there's the top, there's it together. It's kind of like a lid in a pot. That's the best way to think of it. Here's free, there's attached, free, attached, free, attached. This is ER. This ER would be I don't see any smooth ER here, so that's all going to be rough ER. And it's transmitting electron microscope, means we're not making it up. And here's kind of a picture of it. This is the nucleus, and this is the ER. Okay, next one's cytoskeleton. Next one's cytoskeleton. In the phospholipid bilayer, it was those little lines down there. Cytoskeleton is on the inside of a cell that gives it shape and helps in division. And then it's on the outside of cilia and flagellum. Okay, got it? All made out of protein. All made out of protein. There's 3 of them. Here we go. It says proteins organized cytosol. All 2, who cares? 3 cytoskeletal components. You ready? They fall into 3 categories. Here we go. You know it's going to be a test question. Which is or which is not? Here we go. Microfilaments, intermediate filaments, and microtubules. Okay? Microfilaments, mostly we see these as actin, A-C-T-I-N, and myosin, M-Y-O-S-I-N, in the sliding filament mechanism, making a muscle contract. That's where we see most of the microfilaments. When we get the muscles, I will lay that out for you. Microfilaments, intermediate filaments, and microtubules. Ready? Here we go. We're going to take each one of those, talk about them. Microfilaments, composed of actin, I just said that, actin and myosin, intertwined, doesn't make any difference about the intertwined, and myosin. Now there are other ones, but those are the 2 big ones, and they're found obviously in muscle contraction. Okay, maintain and change the shape of the cell. Yeah, they have a cell out like this, and it goes like this because you just contracted it, and the microfilaments are what does it. I'm not going to go anything deeper than that right now, but when we get to the sliding filament mechanism, I will explain it, and I would hope that you would get it. Just about everybody does. Okay. Muscle contraction and cell division. Okay, muscle contraction, also in cell division. Remember mitosis? Okay, here we go. Intermediate filaments. Remember when we said proteins reach out there and hold on to other cells so we have tissue? Remember that? And I said we get a little bit with sugar, the glycocalyx, we get a little bit with that little arm. Remember those green arms that were sticking out there? Okay, we get a little bit of some sticky sticks to hold it together, but the big thing that holds it tight is proteins. The protein is intermediate filaments. Microtubules, microtubules. Next one. Don't worry about that. Tubular protein, don't ask that. Remember the centrosome? Remember I said it looked like gears in that prototypical cell? I said when they're together, when the two are together like this, they're called a centrosome. When they split apart, they're called centrioles. Remember that? And that's, I said that's what anchors, goes to opposite poles and anchors so a cell can be split into two. I will do that for you. Fix organelles. The organelles are fixed in place too. That isn't the only thing it does. Microtubules are also involved in fixing the organelles. You know your organelles move inside of a cell. Okay, they just don't sit in one spot for their whole life. They move around, and the microtubules are what helps them do that. Shape, rigidity, cyclosis of cytoplasmic stream. That's what I'm talking about. When those proteins change their shape, okay, and they change the shape, they move the mitochondria around. They move ribosomes around. They move lysosomes around. Otherwise, lysosomes and all those other things aren't going to have the ability to come into contact with what they need to come in contact with. Makes sense? Microtubules does it. Motility. Also, you put them on the outside. They're called cilia and flagellum. They allow a cell to move. When we say allows it to move, it's called mobility. Mobility means it is moving. Okay? And cilia and flagellum are what do that. I told you flagellum is only on a sperm cell. Blah, 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 blah. If the cell is attached, it can't move. It moves things past the cell. Good? Good. Okay, cell division. Moves the chromosomes in cell division. We're going to call it spindle fibers. I will show you. Centrosome, centrioles. Here we go. Centrosome is when they're together. Centrioles is when they're apart. They're at 90 degrees. They're sets of 9. They make a big deal out. 9 and 2, 9 and 0. Don't worry about it. Okay? I never did. I got where I'm going. Okay? But when you get into cell and molecular biology, they go into a big deal about that stuff. Okay? And they should. Okay? But right here, you don't need to know that. Centrioles when they're apart, centrosomes when they're together. Okay? Involving microtubules. Pictures. See them at 90 degrees? See how they look like sprockets or gears? Okay? When they're together, they're a centrosome. When they're apart, they're a centromere. I mean, sorry, but when they're together, they're a centrosome. When they're apart, they're a centriole. Good. Cilia and flagellum. Outside, capable of moving things or moving themselves. Cilia. Okay, respiratory tree, oviduct. I showed you a picture. Remember, it looked like a sea of grass. Flagella, longer than a cilia. Okay, it's single, used to propel a cell. Now there's some very primitive organisms like a sponge and stuff that the flagellum move things past them. It's not like a sperm cell that moves the cell. They're attached to other ones they can't move, and they make stuff move through the sponge very quickly. Okay. All right, sperm in us. Pictures. Remember this guy? Remember this guy? Scanning electron microscope, 3300 magnification. And this was the picture of it. This is a goblet cell. I'll talk about that later. Okay, and there's a sperm cell. Microvilli. Microvilli. Remember, this is only on the villi in the stomach. Remember, we wanted to increase surface area. You put it in villi. You add villi to it to increase the surface area. And to slow the stuff down, you put in microvilli. Yeah, microvilli. Not capable of motion. To slow down digested substances. Remember, your book is going to say increase surface area. Don't look for that. Don't look for that. Look for slow it down. Absorption of nutrients. Okay, now read that. Okay, I'm not going to read it to you. Read it because what it says is, hey, it's going to slow it down so it can be absorbed by the cells. Okay, remember the third? Remember the third? Here we go. We did the plasma membrane. We did also called the cell membrane. And then we did cytoplasm and all the junk that went into the cytoplasm. Now we're going to do the nucleus, just about the nucleus. Control center, the three major components. Okay, here we go. Test question, three major components. Nuclear envelope, remember it had the holes? Well, what else do you know about a nuclear envelope? What about an envelope? Does it have two sides or what? It has two sides because there's a pouch in the middle. Okay, remember, double membrane. I said there was two things that were double membrane. You remember that? Mitochondria, the phospholipid, and the nucleus. Nucleoli. Now, here I would be able to use the example in lab. When you looked under the microscope at a cell, you saw a dark spot in the middle. And you said, ah, the nucleus. No, I would tell you to tone down your light, and you would see there was a lighter circle around that dark circle. The lighter circle is the nucleus. The dark circle is the nucleolus. Guess what? That's where ribosomes are made. Remember ribosomes for protein synthesis? Well, they're made someplace. They're made in the nucleus on a nucleoli or nucleolus. Okay? Plural singular. And a cell may have more than one. Okay? And they're non-membrane bound. DNA, chromatin, and chromosomes. I have to take a minute for this. Most people don't understand this. I'm going to use that pearl necklace. Okay. Chromosomes and chromatin, same thing. Okay? It's a pearl necklace. Now, that pearl necklace in a normal cell being read and telling what kind of RNA to make and doing all that code and all that other stuff is called chromatin. Okay? It's only called chromosomes in anaphase of mitosis. Only during anaphase of mitosis. Okay? Now, people say it wrong all the time. Don't get into a big debate with somebody because they don't care. So if somebody misuses the word chromosomes, don't get all, you get your undies in a bunch. Just say, obviously, they don't know what the hell they're talking about. No, it's called chromatin, unless it is in anaphase. Only time it's called chromosomes. You get it. Now, we all refer to that as DNA. Here's the way I want you to go to a pearl necklace. You have a pearl necklace. It is called chromatin in a regular cell. In a dividing cell in anaphase, it is called chromosome. We have 46. I'm going to use the word chromosome right now. We have 46 chromosomes, 23 pair. You get 23 of them from mom, and you get a complementary pair, 23 pair from dad. The two come together, and now you are a diploid cell. Okay, don't worry about that. I'll tell you about that. We do meiosis. Regardless, every pearl is enough genetic information to make a trait. So to make these blue eyes was one of those pearls. Okay? To make insulin was another of those pearls. And those pearls are put on specific spots on specific necklaces. And you have 23, well, you have a total of 46 necklaces, 23 from mom and 23 from dad. And let's say necklace number 7, pearl number 212 is the same thing as number 7, 212 for dad. And then you have the dominant recessive, which we won't get into. You can do that when you do general biology. Got it? Got it? Now, if we take that pearl, which is enough genetic information to make a trait, and we smack it with a hammer, it breaks open, and we're going to find out it's made of nucleotides. Okay? And nucleotides are what DNA is. Okay? Nucleotides come together in a certain code, twisted code, called the nucleic acids. And there are two types of nucleic acids, RNA and DNA. Okay? So what happens is you take nucleotides, they're like the building blocks, and you take a certain amount of them together and you get a gene. A gene is enough genetic information to make a trait. Chromatin and chromosomes are made of string of genes, pearls. What is the pearl made out of? DNA. You understand? Let's do it one more time. Starting from the smallest, get to the biggest. Nucleotides form nucleic acids. There are two types, DNA and RNA. Nucleic acids, enough nucleic acid to form a trait is called a gene. Genes strung together are called chromatin or chromosomes. Got it? Most people don't get that. Okay? So DNA is only a piece of the puzzle. All right. DNA stands for deoxy. RNA stands for ribo. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Just drop the deoxy. Good? All right. Double membrane, we did that. The holes are called, not called holes, they're called pores. And since they're in the nucleus, they're called nuclear pores. Remember ER? Well, the outside of the envelope of the nucleus is continuous. It means it kind of continues out and forms the endoplasmic reticulum. Good? I think I asked it. Let me think. Oh, I got my water. The only problem is it means I'm going to be up all night getting rid of it. I think I did. Nucleolus. Nucleolus, nucleoli. Plural, singular. Got it? What did I say about that? Can there be more of one of them in a nucleus and a nucleus? Answer is yes. What do? They make ribosomes. That's what ribosomes are made of. Membrane, unmembrane. Membrane, non-membrane. Non-membrane. Nucleolus. Composed of RNA, enzymes, various proteins responsible for making components of a ribosome. That's what they're made. Ribosome formation. That's what it's going to be on your exam. Ribosome formation. Nucleus. Picture. Okay, let me show you what you got. See all those little things? Those are ribosomes. See that? Nuclear pore. You see that? One inside layer, outside layer. Double membrane. You see that? This is your chromatin. This is nucleolus. It only shows one of them. Okay? Remember, continuous with endoplasmic reticulum. See, it kind of comes out and forms an endoplasmic reticulum. We good? Okay. Picture. DNA, chromatin, and chromosomes. Now, I told you this. We're going to go over it specifically. The nucleus houses deoxyribonucleic acid, DNA, your genetic material. Okay? Who you are doesn't change. If it does, it's called cancer. If you don't change that DNA, got it? You don't change that DNA. You started off as one cell. Your mom's oocyte, egg, met your dad's sperm. Says, how do you do? They joined together. 1 + 1 = 1, and you had one cell called a zygote. And that cell divided and divided and divided and divided and is still dividing now. You're made of 1 trillion of those one simple single zygote. Okay? Now, that doesn't change. That genetic material does not change. Now, realistically, does it change? Yeah, because your exposure to all kinds of stuff. Does it change? Yeah, it's not supposed to. And we hope that it doesn't. And sometimes if it changes in the right place, it triggers cancer, okay? Anyway, that's in cell and molecular biology. Chromatin says when a cell is not dividing, in other words, doing what it's supposed to be doing, those pearl necklaces are called chromatin. It says during a dividing cell, specifically anaphase, it's called chromosomes, okay? Still a pearl necklace. Now, it says when there's enough genetic material, nucleotides, see it? Enough genetic material, nucleotides, nucleic acid, to make a trait, it's called a gene. That's a pearl. Got it? So let's take a look at this. This is your nucleotide is composed of 3 things. Nucleotide is composed of 3 things. A phosphate, a sugar, and a nitrogen base. You've heard of this. In DNA, there's 4 nitrogen bases. T's and A's and G's and C's. Thymine, adenine, guanine, and cytosine. Those are the 3. I think you see them written right here. Thymine, let me get my little arrow. Thymine, adenine, cytosine, guanine. Got it? And a nucleotide is made of a phosphate, a sugar, and a nitrogen base. So a phosphate sugar in one of those. So here's your phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And off of the sugar comes the nitrogen base. And And A's only recognize T's and they make a bond. And G only recognizes C, and that forms a bond. A to T has 2 hydrogen bonds. G to C has 3. That's why you see those little dots there. I'll get to it. There it is. There it is. See how there's 3 and there's 2? Okay. 3 and 2. Okay. That's just a fact. That means you can never get your genetic code jacked up. They can only bond one way to another. Okay. Now, in the old days, like in the late 40s and early 50s, they used to think your genetic material was proteins. Okay. And they said, let's separate it. Let's boil it all up, mash it up, blend it, and we'll see what we get. We can check for proteins. We know how to check for proteins. So they did. They took the nucleus out, blended it in a blender, checked it, and guess what it was? Proteins. They didn't know what to check for. They knew how to check for protein. They didn't know how to check for anything else. That's because your DNA is wrapped around proteins called histones. You see the histones? So it's wrapped around there to keep it from getting mixed up. And that's what they were checking for. And it wasn't until 1953 that these 2 dudes, one from the United States and one from England, you probably know them as Watson and Crick. Okay? Watson was from Loyola, I think, down in LA. And Crick was from England. And they came up with the structure of DNA. Okay? And they got the Nobel Prize for it in 1966, I think it was. Anyway, they're the one before that. There was 3 macromolecules: proteins, fats, carbohydrates. There was no nucleic acid. Nobody knew about it. And they're the ones that said, hey, and they were right. And they discovered this part right here. Okay? So a nucleic acid is made out of building blocks. The building blocks are called nucleotides. Got it? A nucleotide is composed of 3 things: phosphate, sugar, nitrogen base. And there's 4 nitrogen bases in DNA: thymine, adenine, guanine, cytosine. And guanine and cytosine always match up. Adenine and thymine always match up. Now, for you people that have had some cell and molecular biology, you know that, oh, but in RNA, something's different. Yeah, you take the T, throw it away, and you put a U in there. The G and the C don't change, and they form 3 hydrogen bonds. The T and the A changed. T's gone, U takes its place, so it's A and U with 2 bonds. Okay? So in totality, there's 5 nitrogen bases: adenine, guanine, cytosine, thymine, and uracil. Good? Wow, you're learning all kinds of stuff. Now, what's nice about you is you can turn off your computer and go get a drink of water or run around the block, clear your head, and come back. I got to keep doing this. I'm getting tired, and I'm guessing at all this stuff. Okay. Now, remember the last thing was the life cycle of the cell. Now, we're getting towards the end of this. And this is where we have mitosis. And the end of mitosis is the end of the presentation, and the next one I do is on a board. And that's it in my lab. I'll go in my lab, and I'll have a camera on there, and I'll have my lab coat on. I'll be doing all kinds of stuff writing on the board. Life cycle of a cell. Okay, all cells in your body are in a cycle, birth to death. You understand? Some of them are in the cycle longer than others. Some are in different parts of that cycle longer than others. A nerve cell might be in it for its entire life and never go through mitosis. And something like a white blood cell may be in it very quickly. In other words, if you have an infection, your white blood cells are undergoing mitosis. They're going under mitosis like you can't believe. You understand? So the best way to do a cycle is a circle. Okay? There's 2 states. Now here's what you're going to want to remember. 2, 3, 4. In fact, 1, 2, 3, 4. There's 1 cell cycle with 2 components: interphase and mitosis. Interphase has 3 components: G1, S, and G2, and mitosis has 4 components: prophase, metaphase, anaphase, telophase. Easy way to remember it, the way I teach it: 1, 2, 3, 4. figure those out and you'll know the cycle. Okay, now I'm going to go through those right now. There are two states of the cell cycle, interphase and mitosis. Oops. And mitosis. Got it? Those are the two, interphase and mitosis. There should be three blue things here. Normal metabolic activities, put another bullet right here and put DNA synthesis, making of DNA synthesis. There should be three of them, okay? Okay. Oops. I'm looking at this thing. I get this area I live out in a county. And here's the thing. It says dead cow. Be careful when you go by this certain street. There's a dead cow on Borden. Okay. You see a dead cow, I see steak. Okay. And then when mitosis is when the cell's divided. Okay. So what we'll do is we'll go through, I'll draw you a circle. You'll see all these things put up in a circle. And then I'll do, go through interphase and what's going on there and what's going on in mitosis. It's really straightforward, but you do have to memorize it. Interphase. Most of the cells are in that most of their time, okay? So let's say you have a skin cell. Let's say you have, skin cell. It takes about 2 weeks for that skin cell to be born and then to die, okay? About 2 weeks. So it's in interphase. Most of that 22 days, 21 days, it's in interphase. And then the last day it undergoes mitosis so it can be replaced. You understand? Okay. Divided into 3 stages. Remember? 1, 2, 3. 2 is interphase mitosis. 3 is the stages of interphase. Here we go. G1. That's normal cell growth, replication of organs, produce proteins for replication, centrioles, that kind of stuff. G1. S, synthesis, DNA synthesis. Remember when I told you to add that one? So you synthesize DNA, you make it, you copy it, you clone it. Got it? And G2 is preparation for cell division. You start adding some proteins in there and you start getting ready to go through prophase, metaphase, anaphase, telophase. Do you understand? Okay, that's interphase. Centriole replication is complete. Here we go. Here we go. Okay. Enzyme cell division. Enzymes are synthesized because everything in your body is under the influence or done or capable of being done by enzymes. Enzymes do it all, Jacko. Got it? Okay. Those are made during G2. Cell cycle picture. What I like to do is I like to draw a circle and then put a cross in the middle. That divides it into 4. Okay, now you look at here. See where it says mitosis? Here, wait for it. See it says mitosis there and it says interphase. So if we do a straight line, the cell cycle would be divided into interphase and mitosis, 1 and 2. How many cell cycles are there? 1. It's divided into 2, interphase and mitosis. And we know interphase is divided into G1, S, and G2. G1, normal cell growth, doing what it's supposed to be doing. S, DNA replication, synthesis. G2, don't look at growth, they ain't right. It is preparation for cell division, making the enzymes and that stuff. And then it goes into mitosis, prophase, metaphase, anaphase, telophase. No cycle. Cytokinesis, no, it happens during telophase. That is not supposed to be there, it's supposed to be in there. And what this is showing you is what it will look like under a microscope. And we have a lab where you'll be able to identify this. The easiest one, easiest one. See how all the sister chromatids are lined up in the middle? Middle, equator, yes, middle, metaphase, middle, metaphase. Good. See how they just break apart? Break apart, anaphase. Guess what those are called? Come on, I just told you. What are they called right here? Chromosomes. Good, you got that right. And telophase, you do the figure 8 and you break them off into 2 cells. Yeah? During telophase, you have cytokinesis. This does not go there. That's wrong. Okay? There we go. Let's go back into it. Interphase, mitosis, and cytokinesis. Okay, do you see this centrosome is, the centrosome has split. Those are called now centrioles. You see that? The nucleus is still there. That means all your chromosomes, your chromatin, your DNA is all 46 of those guys are still in there. Don't worry about the nucleolus. Don't worry about them. They're all in there. And this is what it would look like under a microscope. This is interphase. The way a cell normally looks, except for that guy right there. Okay? Which means it's heading there. It says mitosis. Here's what I want you to write. Here's what I want you to write. Ready? Mitosis is cell replication. Cell replication. Genetically identical. So it's like cloning, huh? Got it? That's what I want you to write. It's going to be exactly, the mother is no longer there. Mother gives herself up to two daughters. They're twins. Mother's gone. Mitosis, division of the nucleus. Okay? I want you to know cell replication, not division of the nucleus. That's what's going on. You're dividing up the nucleus. Cytoplasm, no big deal. That's in cytokinesis, and that's why I put it in blue here, in capitals. Happens during telophase. Cytokinesis happens during telophase. It does not happen after. It's kind of like a no-brainer. If you got a plasma membrane wrapped around you, how can you shift one clump of cytoplasm from one cell to the other? It won't go that way. Sorry. Okay. So cell replication, mitosis, division of the nucleus. That's what's happening. That's where your DNA is. Stages of mitosis. There are 4. Ready? PMAT. P-M-A-T. Prophase, metaphase, anaphase, telophase. We good? We good? That's it. Is that a test question? Yes. Is that a very difficult test question? I would hope you would say no. Okay. The cell cycle, another picture. So what I did is we talked about these guys, and then we just did this: prophase, metaphase, anaphase, telophase. Cytokinesis is inside of telophase, so that's not there. And we said that when the sister chromatids are lined up in the middle, metaphase, metaphase. Pull apart, they're also called chromosomes. They're not sister chromatids anymore, they're called chromosomes. They pulled apart, anaphase. And you see the furrowing, that's what it's called, invagination or furrowing. And that happens in telophase until the nucleus reforms and you got 2 new cells. Okay, good? Prophase. Okay. I have these outlined in the lab for you, the lab that we're going to do on mitosis. I'm not going to read this to you. That's slapping you in the face. But we said that they're supercoils. Remember that? In prophase. And they're wrapped around histones. Remember that? So we don't lose anything. Okay. And we said it's only called chromosomes in anaphase. Okay. What we do is we take this DNA that's kind of stretched out like limbs on a tree, and we start to twist it and compact it so we don't lose any. You don't want to go, oh man, there went my eye. Okay. You don't want to lose any of your pearls, so you got to wrap them up nice and tight. And they're called, when you duplicate. Now you remember that string of DNA we had? Yeah, a string of chromatin we had, 23 from mom, 23 from dad. Well, they're going to replicate. So you're actually going to get 2 moms and 2 dads. Okay. When they do that, 2 moms and 2 dads, they're called sisters. Mom's no longer there. Mom had twin kids called sister chromatids. Right? And they're held together by a protein called a centromere. So, I don't know if you can see my finger. I got one finger here, one finger here. Okay. This was, let's say mom's. This was mom's chromatin number 7. And it duplicated in S phase. Now they're held together right in the middle of my knuckle. They're held together by a protein called a centromere. And they're not called chromatin, they're called sister chromatids. Sister chromatids. Okay? Okay? Now you get spindle fibers. This is all in prophase. I'm listening. You have spindle fibers. Remember those microtubules? They attach to a kinetochore, which is on the opposite side of the centromere. So remember when I did this? Well, right here is another little protein called a kinetochore, spelled with a K-I-N. I'll show you what that looks like in a minute. And the spindle fibers attached to it, and they anchor to, remember that thing called a centriole? It was a centrosome? It anchors to that. And then what they do is they start pulling like you stretch, like you're contracting a muscle, and it snaps that centromere. And one sister goes one way and one goes the other, and you wrap a nucleus around it, you got 2 identical cells. Okay? Nuclear envelope breaks up in prophase. I'm going to ask you these. What happens during prophase? There you go. Bullet. There's 4 bullets. Memorize them. There's a picture. Looks like scrap, like spaghetti right in there. Metaphase. Remember that? Meta, middle. They line up in the middle, huh? So when they're moving to the middle, it's still prophase. Once those sister chromatids get to the middle, it's called metaphase. It says chromosomes line up along the equatorial plate. That means equator. Okay? That's when the spindle fibers attach to the kinetochore. Got it? Got it? See them lined up in the middle? Now here's a photomicrograph. Do you see them lined up in the middle? Wow. Yep. There's the little spindle fibers. These are called asters. So these are spindle fibers here. Those are your centrioles. What you don't see is asters. That's anchored to the plasma membrane, so it can pull it. Anaphase. Ah, remember anaphase? Chromosomes. Yes. The centromere breaks. There's it breaking. Do you see it? Spindle fibers. You still don't see the aster. You still don't see the aster. You see the centrioles. You see the chromosomes because they're not sister chromatids anymore, and they're not chromatin. They're chromosomes. Okay. And then we see the furrowing. Just a minute. Just a minute. Where am I at? Hello. Hello. Right there. That's furrowing. Okay. You see them break here? These are your chromosomes. Got it? Then we go into telophase. Sometimes people call it telophase. Telophase, the nuclear, opposite of what happens in prophase. The nucleus reforms. The spindle fibers disappear. Remember the cleavage furrow, the furrowing, cleavage furrowing? Cytokinesis occurs here. The asters disappear, and the centrioles move back into a centrosome. Opposite of what happened in prophase, huh? See it? See the furrowing? Here's what it looked like. See the nucleus reforming? Kind of hard to see the nucleus reforming here, but it is. I promise. Aging of the cell. Oh boy. Your cells get tired. Do they wear out? Do they kind of go bye-bye? And the answer is yes, they do. Aging of a cell. I have some terms for you. You ready? Aging is a normal and continuous process. What do I know that? I wish I had my 20-year-old body back. I abused that thing, man. I did stuff. I jumped off roofs, jumped off trees, I did all kinds of stuff. Jumped out of airplanes. Went scuba diving with sharks. I did all kinds of stuff. Now I look back at my whole poor body is all scarred up and it's tired. When the alarm goes off in the morning, my body says, ignore it. Okay? But you got to keep pushing yourself. So the better shape you stay in, the higher, better quality of life you'll have for the rest of your life. Even though you're going to get older, you're going to slow down. Okay? If I could take away these wrinkles, I'd look like I was 20 years old. If I could grow hair, I'd look like I was 20. I would love it. I would not mess it up like I did the last time. Anyway, aging is normal and continuous. What ages? Not you, your cells. Okay? 2 general ways. Ready? Terms you're going to see on an exam. Necrosis, you don't want that. That's abnormal or accelerated cell death. That's disease, toxins. Have you ever smelled necrotic tissue, dead tissue? You're going, oh man, something's dead around here. That's the proper term is necrotic tissue. It's dying too fast. It's producing toxins, and you get that stanky odor. Okay. Then normal cell death is called apoptosis. Apoptosis. But you see how it's spelled? Apoptosis. That second P you don't pronounce. So it's apoptosis. Programmed cell death. And your cells have a programmed death. Yeah? Okay. How was that? I'm exhausted. Okay? So this is only part of your day, right? You still have to do a lab. You have to do a mitosis lab. You have to do all that kind of stuff. If you get ahead, have yourself some, oh, I threw it away. Have yourself some sparkling water or just water with ice in it. And do the next one we're going to do, which is meiosis. Okay? Meiosis, making of the sex cell. Okay? So with that being said, I'm going to stop the recording and post this for you. Okay?